morning to all my cute little champions. Let me see if at all I'm audible as well as visible. Just a second. Perfect. Good morning Pooja, and good morning Amina. Good morning, and Sagar. I think the time has been changed from yesterday due to some reasons. I'm not very sure for what reason, but yes. Indeed, is it a good morning for everyone out there? So we shall wait for some more time so that all my dear children can also join. And I hope we all machas and machis are super ready for the most important chapters out there, that is principles of inheritance and variation, and also molecular basis of inheritance. Good morning, Mir. Good morning, Mir Jafar. And make sure, dear children, that you share this video link with all of your friends so that they can also join. And yes. Yeah? Everyone super energetic. Hello Pooja. Hello Sanjana. Good to see all of my dear children who were constantly there in the previous live as well. Very good morning to you. I hope I did not disturb either of your sleeps. Okay. So shall we start? We shall start. Let others join one by one, and if at all you have your friends who are preparing for KCET, make sure you ask them to join this. This is an important chapter, okay? Kavita, hello. Sanjana, no dear, I do not usually have breakfast. So yes. Hello Sanjana. Hi. Okay, so guys, I hope we are ready, and these are the chapters that we're gonna do, that is principles of inheritance and molecular basis of inheritance. The previous year, the maximum number of questions in biology came from the chapter principles of inheritance and variation, and overall, if at all you see a streak, it has been always 4 questions or 5 questions throughout these many years. Same goes with molecular basis of inheritance. The previous year it went down to 3 questions, but if at all you check back the other years, 5 and then 4. So we can expect that to come back again. So obviously it is super important. Sanjana, obviously I do remember you. I hope you are. There are so many Sanjanas out there. So if at all you are the one that I met over here, hi. And if you're not, still a big hi. Okay, so hello Taste Quest, Dinesh Reddy and Santosh, Shruti. Hi ma'am, this is your first class, is it? So you'll have fun and you'll study a lot. Okay. Okay, so let's go ahead then. Dinesh, Baslingappa and Gautam, Sanjana, so you're the same person. That's so great. Okay, so starting with the first chapter that we have is principles of inheritance and variation. If at all we see basically what exactly are the topics that have given most number of questions. So it has been scattered throughout, but still if at all we have to choose, it would be inheritance of one gene and also genetic disorders. From here you can see lots of questions have been coming, 3 questions, 2 questions and stuff like that. So imagine a place where 5 questions are coming from this chapter and 3 of them from the same topic. Obviously that means that this topic is super important, right? Now similarly over here also you have seen that from here and there, from pleiotropy you are getting question, from sex determination also you have got question, from inheritance of 2 gene also you have got question, from Mendel's law of inheritance as in the introduction part also you have gotten certain questions. That means that we cannot obviously ignore anything, but still, okay, works. Okay, so it's my first class. Amina says it's my first class. I hope you enjoy it, dear. And yes, so let's go ahead now. And what is JJ? JJ. My name is JJ. My name is Jasmine Joy, and the short form is JJ. Let's go ahead with the first question out there. And my dear children, how do you answer it? 1 A, B, C, D. That's how you answer it. Okay, let's go ahead. So identify which one of the following given pair of options is correct with respect to Down syndrome and also Turner syndrome. So over here you have got the the symptoms of Down syndrome and you have got the symptoms of Turner syndrome. You have to make sure that both the symptoms of this disease and this disease is right. You have to match those things. Okay. Hello Anita. Hello. Good morning. Let's go guys. So over here, Down syndrome, short-statured individual. Turner syndrome, gynecomastia in man. Okay. Darshan says the answer for this question is B. Okay. B is what? B as in B option, or are you looking over here? Make sure guys you give me the answer like this over here. Okay. C. GS World says C. Yes, I gave this question in one shot. Exactly. Sanjana says C. Gautam says C. Anita says B. Okay. Amreen says C. Most of you guys are saying C. And C is what? Let's see what is C. So let's go one by one. Short-stated individual. Yes, that is a feature of Down syndrome. But gynecomastia means presence of breast, and that too in a man. That is a feature of Klinefelter syndrome, not Turner syndrome. So this option is wrong. Round head and partially open mouth. This is a symptom of Down syndrome. But overall masculine development, again, this is a symptom of Klinefelter syndrome. They are usually having masculine body, but they also have breast development, right? So this is again wrong. Broad palm, physical and mental development is retarded. Yes, that happens in Down syndrome. And in Turner syndrome, sterile females. Are they females? Yes, they are females, but in them, instead of having XX chromosome, they lack one X chromosome, so it's XO condition. So which is why they will be sterile, because their ovaries are rudimentary. They are not fully formed. Okay, so over here, sterile females with rudimentary ovaries would be absolutely correct for Turner syndrome, and this is also correct for Down syndrome, which is why your option should be C itself. Additional copy of X chromosome, that is not the case of Down syndrome. You have an extra autosome, not a X chromosome. X chromosome is a sex chromosome, right? Absence of X chromosome, that is correct. So all of you who got your answer correct, kudos to you. Okay, and I'll be reading out your names, and before going ahead with that, you go ahead with the next question, that is this. In a dihybrid cross between a true breeding round yellow seeded and true breeding wrinkled green seeded bee plant, the ratio of segregation of round and wrinkled seed traits in F2 generation is. What do you think is the answer for this? And my dear children, please give me the answer like 2 A, B, C, D. That will be really good. Okay, give me the number also. So let me read out. So GS World and Swapna and Gautam, Sanjana, Amreen and Sanjana, other Sanjana are there, Syed, Taste Quest, Venkat, Dinesh Reddy, Mir Jafar, Hina, Abhi, Venkatesh, everyone gave me the correct answer for the previous question. RG Patil also, Pooja Pujari also, everyone, super good. Now we are going ahead with the next question out there, second question. So most of you guys are saying 2C is the answer, and Baslingappa is the one who says it is B. So let's see whether it's 2A or B or C or D. Listen to me very carefully. You are doing a dihybrid cross between what? Between round and yellow seeded plants. Okay, and what? True breeding wrinkled and green. True breeding means it will be like this. Wrinkled and green, this is true breeding, this and this. The pea plant, the ratio of segregation of round and wrinkled seed trait in F2 is, basically you're just talking about these traits. You're just talking about the round traits and the wrinkled traits. And you're talking about what? The F2 generation. Listen, my dear children, if at all you remember, Mendel's one-gen inheritance cross would be something like this. It was also like this. 2 dominant traits, 2 recessive alleles over here. In the first generation, what did you get? All of them were like this. Similarly, in the first generation, what will you get? All of them are going to be like this, right? In the next generation, how was the case? In the next generation, Baba, you saw something very, very important. That is, you saw a ratio like this, where basically 3 of them were tall. Why tall? Basically, these are tall because you have one dominant allele and this is automatically tall. So 3:1 ratio is what you get for tall and dwarf. Similarly, for tall you do over here, you will get a similar ratio itself, no? Over here, if at all you see, it is going to be like this. 3 of them, Baba, are going to be what? Round. And 1 of them is going to be what? Wrinkled. So your answer for this question is going to be B, right? Your answer is going to be B. Okay? Yes? So many of you got it wrong, which means that there is a problem. Now tell me the problem, and then only we'll go ahead with the next question. Because is there an understanding problem, as in why it was there? And there's an easy way to solve it, by the way. Okay, if at all you remember the dihybrid ratio of Mendel, okay, it goes like this. 9:3:3:1, right? We say here 9 of them are going to be round and yellow. That is a dominant character of the parent, right? 1 of them is going to be wrinkled and green. Here also 3 and 3 of them are going to be new parental characters, not the older ones, it's new characters out there. So instead of round being with yellow, this time it is going to be with green, and wrinkled over here being with green, now this time it will be with yellow. And over here, if at all you see 9 plus 3, that means basically I can say 12 of them are going to be round, and 3 and 1, that is 4 of them are going to be wrinkled, if at all you're just talking about round and wrinkled. And if you do this, you'll get 3:1. Got it? Question made us confused. Yes, this question is in the masterstroke video. Definitely you will be spotting questions from the masterstroke video also, because that was meant to be for KCET, right? For KCET and PU2. Definitely. Clear? So all Macha and Machis, is it clear? If it is clear, can you just quickly give me a thumbs up? Quickly give me a thumbs up. Yes? Perfect. Then we'll go ahead with the next question. So Vineet, Sanjana and Amina, Tippeswamy and GS World, everyone going good. Okay, Swapna, Pujari, RG Patil. And by the way, if at all I'm missing out any of the names, I'm so sorry. Okay. Ma'am, you provided a video of how to manage 10 hours, but there's a problem. If you follow the schedule, then our daily challenge will miss out. How to manage, tell please. So basically, it's not like so when we say you have got enough time in between, obviously you would have to spend extra amount of time if at all you're spending it over here, right? So you have to manage in that way. Probably you might have to start off early. You can change your time slots. It's according to you how you're going to prepare. So for example, basically you are preparing for biology only in the morning. If at all you go by our timetable, 2 subjects a day. If at all you're preparing for biology in the morning, then pick up those chapters itself in that particular slot of biology, which is going to be there in the live, right? Similarly, if at all there is mathematics in the other half of the day, then pick up those chapters which are there going to be there in the live for mathematics. It will definitely help you out. Yeah? Yes. Perfect. Thank you so much, dear children. Going on really good. Question number 3. Which of the following statement is correct? Listen, this is very important question. Read it out carefully, okay? Which of them is correct? Every time when they are asking you what is correct and what is wrong, what do you have to do, Macha? Over here, you have to underline it, what you're looking for. Change in whole set of chromosome is called aneuploidy. Sickle cell anemia is a quantitative problem. Female carrier for hemophilia may transmit the disease to the sons. Thalassemia is a qualitative problem. What do you think is the answer for this particular question? Okay, which subject is in the afternoon? It will be mathematics, and I think at 5 o'clock. Yes. 3C. Swapna says it is C. Vineet also says it is C. Mir Jafir says C. Okay. Baslingappa says B. Ammu is there. Hello, Ammu. C. Tippaswamy C. Hina Kaushar. Mamata. Hello. GS World. Rutika is there. Hello. And Bhagyashri, Manish. Oh my God, so many children are there. Rangaswamy, Sanjana, everyone are mostly vouching for C only. But I see some people are there confused between B option. Okay, let's check it out. Change in whole set of chromosome is not called as aneuploidy, Baba. It is known as polyploidy, right? In aneuploidy, either there will be loss of one chromosome or gain of one chromosome. There's no whole set of chromosome being changed out there. That is polyploidy, and that is seen in the case of what? Plants. So this particular statement is going to be wrong, right? Yes, that is wrong. Okay, ma'am. Second option. Sickle cell anemia is a quantitative problem. No, Baba. The quality of hemoglobin is getting hampered because you are doing some mutation. There is some mutation happening there. The quality is getting hampered, not the quantity, which is why it is not a quantitative problem. It's a qualitative problem. Now, female carrier for hemophilia may transmit the disease to the sons. Listen, if at all she is a female over here and she has hemophilia, Baba, sorry, she is a carrier for hemophilia and she has an option to give the chromosomes to the son. Obviously, there are chances that she can give this. And from the father, anyways, you have to get Y chromosome to make a son out there, right? Because he is the only one who can contribute. So don't you think a carrier female can pass it to the son? Definitely. So this particular statement would be correct. Now, thalassemia is a qualitative problem. Wrong. It's a quantitative problem. Remember, in alpha gene, in beta gene, there are multiple alleles. Depending on whichever is going to get affected, basically, it can be more effect or less effectful. So it's a quantitative problem, not qualitative, which is why this will also be wrong. So your only answer for this question is going to be C. And everyone who gave me the answer as C, and everyone who did not give me the answer as C, you also learned something new and be happy for that. Okay, clear? Everyone, if it's clear, just give me a quick thumbs up. You know what, in background the prayer song is playing, so okay, so hello ma'am, good morning. Rathod, good morning dear. Phase 1 is PUT, but some teachers are making videos for views, so I have a problem. What should I do? I am not opposing any teacher. Sorry for that. No, it's basically that there might be some children who are struggling with the PU2. They might have not covered PU2 well in the second PUC examination as well. So and it's of high importance. So that's the exact reason, including me, I also started with second PUC more, and then I thought that okay, after this is being done, you are solid, you're foolproof, prepared for it, then probably we can go with PU1. But do not be offended if at all you are going with PU1, please go with that. It's like a brush up. I told you in the daily schedule also, whenever you are doing PU1 also, please spend some time for the revision of the PU2 chapters, otherwise you might forget it. So just think of it as a revision, but you can carry on with the PU1 chapters itself. And then when the PU1 questions come, basically it will be like you are already prepared for the PU1 chapters, right? So it will be easy for you. Humko man ki shakti dena. Yes, it's the same song. Are you able to hear by any chance? I don't know. So the same song that was being playing over here. Okay. Anyways, going with question number 4. Match the column 1 with column 2. So this, again, you have to be attentive. Please know the terms. You might be new to the terms out there, but yes. Autosomal trisomy, allosomal trisomy, allosomal monosomy, cystic fibrosis. I always say in match the following question, read all the options first, then only start off with matching. And when you start off with matching, match these two first. Okay, autosomal trisomy. That means, my dear children, I hope you guys know that basically, what was it? Yeah. So basically, autosomes are the ones which are there in our body. Okay, the chromosomes they are found in most numbers in our body. When we say that we have 23 pair of chromosome, 22 pairs, my dear children, are going to be what? Autosomes. The 23rd pair is going to be sex chromosome. It can be XY or it can be XX. Right? Right. So these chromosomes which are not partaking in any of the sex determination mechanism and things like that, they are known as autosomes. And what do you call the sex chromosomes? You have another name for sex chromosome. It is known as allosome. It is known as allosome. Allosome. Okay. By the way, all of you have started giving me the answer for this question. Oh my God. All the machas and machis are on fire. Darshan says the answer for this question is D. Okay. Now I'll go with your answer. Okay. And Tarun and Anita and Hassan is there. Hello Hassan. Divya is there. Hello, hello Divya. Swapna, Mamta, Hina, and Bhagya, and Tipeswami, Sagar, Pooja, Girish, Vinita. Oh my God. Rutika, Mirjafe, Sanjana, and Sagar. Okay, guys. So let's go off. Over here, let's try to solve it. Autosomal trisomy. In your autosomes, one extra chromosome is coming, that is known as trisomy. And this is what you see in Down syndrome. In Down syndrome, the 21st chromosome, until 22nd, it's all autosome. The 21st chromosome has an extra chromosome out there. Extra chromosome is coming, so that is why it's trisomy. So A goes with 4. You guys are absolutely correct when you said that. Okay. Now cystic fibrosis. Cystic fibrosis is not being explained in your NCERT, but it is clearly written. It is what? Autosomal recessive disorder. It is autosomal recessive disorder, and basically it's a Mendelian disorder. It does not have anything to do with the chromosome number and stuff like that. So basically, I'll say it is a Mendelian disorder. D goes with 2. D goes with 2, and A goes with this. None of the other options have something like this, so which is why my answer will be this. Allosomal trisomy, that means the sex chromosome has an extra chromosome over here. That happens where in Klinefelter syndrome, the guy had to have what? XY chromosome, right? But you have an extra X chromosome right over here. So that is why over here I will say the answer for this is Klinefelter's. Allosomal monosomy, that means the sex chromosomes, one chromosome is lost, and that happens in Turner syndrome. You should have been having XX chromosome, but one chromosome is being lost. So basically, that will be this, which is why your answer is going to be D. And you guys are super awesome. Okay? And Tarun is asking, what are the different names of allosomes? X chromosomes and Y chromosomes. That's all. Cool. Clear? Everyone, super clear for you? If yes, give me a thumbs up. If it's super clear, what is the reason for low sperm count? It can be anything. It can be immunological reasons also, where your own body cells are going to kill your own cells out there. That can be a reason. Or because of stress, or because of taking drugs like smoking cigarettes and stuff like that. It has multiple reasons. Okay. Perfect. Yes, Tarun, perfect. Okay, thank you guys for giving me the thumbs up. Lots of love to you guys, and I really hope you're having a good morning today. And by the way, if still any of your friends are there who have not woke up yet, so you tell them that, hey, there is a class of biology, come and join. Okay. So XO type of sex determination and XY type of sex determination are the examples of what? Male homogamity, male heterogamity, female homogamity, and female heterogamity. This is supposed to be an easy question. Okay, guys, please give me me the answer like 5 A, B, C, D, please, please, please. Okay, Darshan says B. Okay, Divya Prabha, very good. Okay, 5B. Okay, Taste Quest says B. Tippaswamy says B. Everyone, Manaswini is also there. Hello, hello, hello. Everyone mostly are saying B. B is what? Male heterogamety. Why male heterogamety? XO type of sex determination is found in what, Baba? Is found in insects such as grasshopper and stuff like that. If at all you see, this is the female and this is going to be male. What do you mean by heterogamety? That means when they form gametes, in gamete only one chromosome is supposed to go. From here, whatever they do, Baba, all the gametes are going to look the same. It's going to be X chromosome only. But here it can be X chromosome or it can also be no chromosome at all. Two different type of gametes are being produced, right? So this is why it is an example of male heterogamety. Similarly, in our case, in the case of human beings, and what is the other example? Guys, any example do you remember of male heterogamety other than human beings where you see this XXXY type of sex determination? Any idea? Well, in the case of human beings, we say they show male heterogamety. Why? Because two different gametes are being produced by the male. But I am asking you, Drosophila. Exactly. Manaswini says Drosophila. Tarun also says Drosophila. So in Drosophila also you have XXXY type of sex determination. Okay, do not forget it. In insects such as grasshopper, you have this. In human beings and Drosophila, you have this, according to your NCERT. Ma'am, why it is important to solve previous year question papers? Because most of the concepts could be repeated. I just showed you so many questions have come from the genetic disorders. So when you solve questions from genetic disorders, automatically you are becoming pro in that particular topic. Don't you think you will be able to attempt more? Definitely yes, right? Yes, Mamta, Tarun, everyone. Tippaswamy, Sagar, and yes. Birds, dear birds have female heterogamety, not male, not something like this, okay? So everyone who gave me the correct answer for this, kudos to you, and everyone who did not give me the correct answer, I hope you have got an understanding of this. Now going ahead with question number 6. Are we supposed to go? If yes, can you give me a quick thumbs up, my little children? Can you give me a quick thumbs up if at all it's clear? Cleared, Macha. Thank you. Is it? Okay. Yes, only similar concept is going to come. Questions are not going to repeat it. Ayyo, Macha, you only think the people who are writing the examination, if at all they repeat the same question, everyone will get the admission. No. So they don't want that. They want only the children who are super prepared to get the admission. Right? Thank you guys for giving me the thumbs up. Sanjana, Sagar, Hina, Vineet, and yes, everyone out there. Pooja, everyone. Thank you. Let's, did I show you the next question's answer? Yes, I did. But yeah. Anyways, the genotype of a husband and a wife are IA, IB and IAIO. Among the blood types of their children, how many different genotypes and phenotypes are still possible? How many genotypes and phenotypes are still possible? What do you think is the answer for this particular question? Ma'am is paper liquor, is it? Thank you. Okay. B, guys, please tell me the answer in 6A, B, C, D. Don't give me the answer like this. You know what happened one day? So I said the answer is correct, but I didn't know that child is answering for the previous question out there. So don't do that, okay? I will be guilty later on. So Sanjana says 6B. Okay, Vineet says B. 6B. Vijay says 6B. Sanjana, okay. Bhagyashree B and Rutika B. Pooja B. Everyone, most of you guys are saying B, which is why I think we should be, Rathod is also saying B. We should be going ahead with B option. But how B option? Let's see. So you're talking about IA and IB. Clear? Understand this particular concept, okay? Very important. And then you're talking about IAIO. What are you being asked? Different genotypes and phenotypes are how many different genotypes and phenotypes are possible? That is the question. Let's try to do that. This and this goes, I get IAIA. Okay, and we write the phenotype also. What will the blood group? It will be A blood group because both of the alleles are A only. Okay, fine. This and this goes, I get IAIO. Oh, next genotype, new type of genetic nature, but still it will be A blood group only, right? Because A is dominant over O. Okay, ma'am. This and this goes, I get IA and IB. Both of them are going to be co-dominant, which is why the blood group is going to be AB. Okay. This and this goes, I get IB and IO. Again, B is dominant over O. Okay. So it will be B blood group. How many different genotypes are possible? If at all you see, none of the genotypes are repeated. Everything is new, brand new. So basically, I can say 4 genotypes are there. But when I talk about phenotypes, you do not count as 1, 2, 3, 4. No. You do not do like that. Basically, you count it as 1 because they are repeated, and 2 and 3. 1, 2 and 3. So I'll say phenotypes are what? Phenotypes, physical appearance is what? 3 of them, and genotypes is 4. And anyways. So they have given multiple options to confuse you guys. Do not be confused. And I know you will not be, because you guys are what? Super awesome. Is this question clear to everyone? Is it clear to everyone? This is important, okay? Blood grouping is also important. Do not miss it by any chance. Cool? Works? Yes, clear, Macha. Thank you. Definitely, Tarun. We'll be making a separate video on pedigree, and I've involved pedigree related questions over here as well. Chidambar is here. Hello, Chidambar. Sanjana. Okay, thank you, Sanjana and Baslingappa and everyone out there. Okay, perfect. My dear children, have anyone told you you are the sweetest among everyone? Okay. So let's go ahead with the next question out there. Question number 7. Match column 1 with column 2 and find out the correct answer. So column 1 goes like this. First, read the options. Aneuploidy, monoploidy, polyploidy, diploidy. Ma'am, I have not studied this and this. I have studied monosomy. And some children might be going and fusing with a lot of things out there, thinking that it's monosomy. Do not do that, okay? Read the option carefully and then only give me the answer. Rathod, ma'am, you're teaching very nice. Thank you. Is it? Thank you. You're most welcome. And thank you so much. Abhishek, ma'am, you're my favorite. Thank you guys. You guys are also my favorite. Mir Jafir, Vijay, Hina and Divya, Vijay Tales, Manaswini, Rangaswamy, oh my god, Mamta, Darshan, Syed and Sanjana, Sagar, Taste Quest, everyone, GS World, Bhagishri, everyone is saying the answer for this question is C, and none of you have given me wrong answers. Is that what I see? Okay, so you are saying C is the answer. Let me match. Aneuploidy is loss or gain of a chromosome. Loss or gain of a chromosome. This is very easy to spot, so A goes with 2. A goes with 2. I just have one option like that, and with that only I found my answer. But still, let's go with diploidy. Diploidy means basically what? Diploidy means you have 2 sets of chromosome. Diploid. Haploid means half. When you have 2 of them, you said diploid to 2 sets of chromosomes. So D goes with 3. D goes with 3. Now what do you have? Monoploidy. Monoploidy means basically you have only single set of chromosome. That is basically haploid sort of thing. Okay, single set of chromosome out there. Cool? Yes. Now polyploidy means you have basically increase in the whole set of chromosome. Whole set of chromosome increases there. So basically your answer is going to be C, and you all are correct. You all are super awesome. Okay? Yes. Yes. Without knowing answer we can say, because seeing question number B4. I didn't get it. So sorry. Yes, if one answer is easy, no, it is easy. Yes, definitely yes, it is. So those of you who got your answers wrong, I hope it's clear. And tell me, everyone who is watching this video, can you tell me the example of polyploidy? Where do you see polyploidy, Macha? In which organism do you see polyploidy? Everyone. Everyone out there, just give me a second, okay? Okay. See, strawberry, citrus fruits. Listen, guys. Polyploidy, it's common in plants. Yes, that's all. Okay, so you do not have to go ahead with which type of plant is there. Listen, we are not talking about polyembryony. All my dear champions went to that particular zone of sexual reproduction flowering plants. Come back. We are in principles of inheritance and variation, and over here, polyploidy is seen in the case of plants. Plants. P for polyploidy and P for plants. Okay, they come under what? Chromosomal disorders reason. So reason can be aneuploidy, where there is loss or gain of only one chromosome. That happens because of non-disjunction of chromosomes. So when the cell division happens and the chromosome have to separate, no, sometimes one chromosome says that I will not separate, I will be here only. So basically why? That is why it is not able to go ahead and stays together. That's why we get an extra chromosome, or probably there's a loss of chromosome out there, right? But polyploidy is ulta case, where the whole set of chromosome itself is basically doubled out there. There's an increase in that. That is seen in the case of plants. Do not forget it. Okay? Done? Is it clear? If it's clear to everyone, can you give me a thumbs up? Sanjana, triploid, 3N, seedless, watermelons and banana. Yes. Okay. Cleared? Thank you so much. Now going ahead with the next question. A pure breeding pea plant. By the way, everyone have clicked on the like button. If at all you have not clicked on the like button, if at all you have not subscribed to the channel yet, subscribe to it right now and click on the like button as well. When you click on the like button, what happens? It reaches to other children also. That is how it usually works, I think. Yes. A pure breeding pea plant with round and yellow seeds was crossed with pea plant having wrinkled and green seeds. On selfing. So listen to me while reading the question. Read it carefully. Pure breeding pea plant with round and yellow. You're talking about dihybrid cross. Everything related to dihybrid cross should be coming into your head right now. Okay. Okay. Round yellow seeds was crossed with pea plants having wrinkled and green seeds. Okay. On selfing of F1 hybrid. Basically you got the F1 and you selfed also. You got 64 progenies in the F2 generation. Find out the number of F2 progenies showing non-parental character. In the second generation out there, which one are the ones which are showing non-parental character, new character? And you know what? In PU2 examination, I gave this question's example so many times. If at all you are someone who were there for my PU2 preparation, you might already know the answer as well. Hi, good morning, Tasmia. Okay, Manju, good morning, dear, and everyone who is giving me the answer, Baslingappa, Vineet, and Sanjana, everyone out there. Okay, Sanjana, Ratwa, and Katwa, sorry. Hema, hello, dear. 8C, 8A. So there's a confusion between 8 and C. Hassan, yes, gave me the formula also. Kudos. Exactly. So most of you guys are saying 8A, but I feel that yes, there is a confusion. We might not know how to solve it. So listen to me very carefully. Basically, they spoke about a normal cross, a dihybrid cross, and they spoke about the F2 generation. So we have to reach until F2 generation, and we know only one thing about F2 generation. That is what? There is a ratio like this. I told you 9 of them are going to show what? The dominant parental character, that is round and yellow. And 1 of them is going to show the recessive parental character, that is what? Wrinkled and green baba. Right? These are parental characters. Now in F2 generation, you are also able to see non-parental characters, something very new, which you have not seen until now. So what are those characters? Let me see. So round, which was with yellow, is now going to be with what? Green. Do not do rectification, understand this question, okay? Now here also, wrinkled was with green until now, but now it will be with yellow. What are these traits? These are non-parental traits. Did you see these traits in the parents, Baba? No, you have never seen that in the parents. This combination is not seen. So they are non-parental. How many of them are non-parental? 3 plus 3 is equals to what? 6. 6 of them are going to be non-parental. Baba, how many of them are in total? 16. 9 plus 3 plus 3 plus 1 is equals to what? 16 of them. So I say 6 out of 16 are what? Non-parental. And over here I am already given a number. That is, I am saying 64 of them are being produced. It's not 16. It is basically 64 of the progenies which are being produced, and I have to find among them. So basically, if 6 out of 16 were non-parental, and I have to find if there are 64 progeny, what will be the case? I'll just have this particular formula. 6 by 16 into 64. Now, when you do the calculation, it comes out to be what? 6 into 4 is equals to 24. Your answer is going to be A. It is going to be what? A. Yes. So I have tried to include the questions which might confuse in the master stroke. So yes, that means master stroke is important. Don't you agree? Please watch the master stroke videos if at all you have confusion with one shots. Okay. Clear? Good morning, Vinod. Ma'am, you're beautiful. Is it? Thank you so much, guys. You are also beautiful. I hope this is super clear for everyone out there, right? Yes. Let's go ahead with the next question then. Cleared? Macha? Hello, Halesh. Okay, Sanjana gave me thumbs up. What about others? Everyone? Chidambar also gave me thumbs up. That means it is clear. Okay, now on the other hand, if at all you have been asked about parental character, how will you find it out? Basically, 9 plus 1, both of them are parental. 9 plus 1 is equals to 10. So 10 by 16 into 64. Anyways, you do not have to do that. You just do the half. If 24 of them are parental, rest of them will be non-parental. No, anyways. But I'm just still telling you, if at all you had the reason to find the other way, you have to do it. Okay? Ma'am, it's not an issue, right? If at all I call you Macha, it's okay, dear. Okay? Minu, you want me to repeat this particular question? Guys, just give me a second. I will repeat it. Otherwise, it might not stay. So, listen, I'll do it fast. when you talk about dihybrid cross ratio, you are being asked that basically round and yellow are crossed with wrinkled and seed, right? Wrinkled and green color seeds, right? Right, okay. Now in F2 generation, you get a ratio like this, 9:3:3:1. Okay. 9 of them are going to what? Dominant character of the parents, that is what? Round and yellow. One of them is going to be what? The recessive character of the parent, that is wrinkled and green. What about these three things? Sorry, these two things? They are the new character which was not found in the parents. That is why you call them non-parental. What are they? So round was never with green, but this time it is with green. It was always with yellow, but this time it is with green. Similarly, wrinkled was always with green, but this time it is with yellow. So these two things that you see over here are non-parental, and you know that out of 16 progenies, so when you do 9 plus 3 plus 3 plus 1, you get 16. Out of 16 progenies, 6 of them, 3 plus 3, 6 of them are going to be non-parental. And if here you are being given a number. Out of 64, what is the condition? So what do? You multiply it with 64 and you get your answer. Cool? Yes. Correct? Okay. Let's go ahead. Question number 9. In Morgan's experiment with Drosophila, when yellow-bodied, white-eyed female was crossed with brown-bodied, red-eyed male and their progeny were intercrossed, what was the percentage of recombinance in F2 generation? What was the percentage of recombinance in F2 generation? What did you cross over here? Please understand. Yellow body and white-eyed female was crossed with brown body and red-eyed male and their F2 were intercrossed. What was the percentage of recombinance in F2 generation? How much percentage of recombinants were found? Okay, so over here, okay, Darshan says C is the answer, Swapna says D is the answer, Sanjana says B is the answer, Chidambar says A is the answer. That means total I have got 4 options, A, B, C, D. Jasmine ma'am, you only choose the answer because anyway we have different answers for this particular question. Okay, ma'am, say important chapters of 11th and 12th. There will be a video being released today itself, I think so, these 2 chapters are important for 12th, and apart from that the ones that I have already covered. So yes, hair is so silky. Puzzling kappa. Okay, C. Most of you guys are saying C is the answer for this question. So I have to go ahead with C is the answer for this question. Is it 1.3%? Ma'am, I do not understand how are others saying C is the option. Listen to me very carefully. Ayyo. Okay. When you talk about a chromosome, okay, and when you talk about these characters, yellow body and white eye, you need to remember a few things. Yellow body is right up over here, the gene for yellow body. The gene for white eye is right up over here. Okay, these 2 genes are closed, so I'll call them what? Closely linked genes. What do I call them? Closely linked genes. Cool. Now suppose there is a chromosome over here. Okay, and this particular portion from here to here, things are moving from this side to this side. Whatever you do, Baba, whatever you do, these 2 things are going to be together only. These 2 portions might go there, but still they are going to be together itself. Agreed? So do you find any new combinations out there? Very less amount of new combination. These 2 guys are together, only the lower part has changed. The recombination percentage, the new combination percentage is absolutely less. Okay, and when we say less, over here, the least among all of them is what? 1.3%. So I'll say the answer for this question is 1.3% recombination is there. The maximum recombination any gene can show is what? Any gene can show the maximum recombination of 50%. That's the max. Okay, and these 2 are very close to each other, which is why it will be just 1.3%. Got it? This is super important. Do not get it wrong. It's important. Now, on the other hand, if I have a question for you, that is, when yellow body and miniature wing genes are being considered, in that case, what do you think will be the percentage of recombination? What do you think will be the percentage of recombination? Any idea? If yellow body and miniature wing is being considered, not this but the miniature body, 37.2. Yes, it will be 37.2. And I still see most children are not getting it. So listen to me very carefully, okay? Consider this. I have capital A gene over here, capital B gene over here, okay? So easy funda. Small a, small b gene over here. they are very far from each other. Okay, which is why if at all I'm interchanging, what is going to happen? I am going to get a new combination altogether. What is a new combination? Small a comes over here, capital A goes over there. Interchange. But here whatever is there, it remains the same only. Capital B is still over here, small b still over here. Haven't you got a new combination altogether? Haven't you got a new combination altogether when you consider these two genes? Yes. So I'll say these two genes were what? Loosely linked. They had too much space between them. They were loosely linked, which is why the recombination is going to be very, very high. The percentage of recombination is going to be super high. On the other hand, if at all I am considering a case like this, capital A is here, capital B is over here. Small a is here, small b is over here. Now if we talk about the interchange, what is going to happen? Basically, small a and small b comes together on this side, capital A and capital B comes together on this side. Do you see any new combinations being formed? Yes, there will be combinations because the upper part is changed. But here, when you talk about these two genes, do you see new combinations? No. So I'll say that these two genes were closely linked. They were what? Closely linked, which is why the recombination would be what? Less. And there's an easy funda to remember it. I always tell that is you and your friend, if at all you're very close to each other, you're best friends, there is less chance for a new combination to happen. There is less chance for a new person to enter in between your friendship. That means it is closely linked. Less new combination out there. Okay, whereas you and your friend, you're not best friends, there are chances that you can get more friends out there. New combinations can occur. Why? Because you are loosely linked out there. So always remember. So in this particular question, you are being asked about what? Yellow body and white eye. When these two terms comes together, you need to understand that these two genes basically which are there. Just a second. Yeah. So yellow body and white eye, they are very close to each other. So they will be showing only less amount of recombination. And the least that we need to remember would be 1.3. But on the other hand, if at all you have been asked, yellow body and miniature wing gene. Miniature wing gene is present over here. That means they are far from each other. They can show high recombination. And the highest recombination which is mentioned in your NCERT would be what? 37.2%. Maximum actually is what? 50%. But in your NCERT it is 37.2, and the example is yellow and miniature wing. Yellow and miniature wing. Always remember, have you seen the ad for Yes Madam? There's an app, no? Yes Madam. So Yes Madam, yellow body. So yellow and miniature wing, they are far from each other, okay? Far from each other. Always remember that. Cool? Cool? Maximum is 50%, dear. Is it clear to everyone out there? If it is clear, can you just give me a quick thumbs up? If it's clear, quick thumbs up, and then we'll go. Ma'am, how to watch this video in 2x speed? Only after the live you can do that. Okay. Yes, cool. Perfect. So with that, let's go ahead with the next question out there. Question number 10. Question number 10. The reason I am explaining to you it a lot is that you can get multiple type of questions, and I want you guys to face it. Okay? Yes, they will never mention. They will never mention. It's our job to find it out. Yeah? Okay. Now, both male and female have normal vision, though their fathers were colorblind and mothers did not have any gene for colorblindness. The probability of their daughter becoming colorblind is what? The probability of the daughter becoming colorblind is what? Vijay says the answer for this is 10D. Okay. So, Apna says it is A. What about others? Darshan says it is C. Okay. Baslingappa, okay. Hassan says it is 50%, that is D, okay. Okay, C, Baslingappa, C, Venkat says C, Sanjana says D, Chidambar says A, Hina says A. Okay, multiple answers, Syed. Okay, guys. Okay, so over here, both male and female have normal vision, though their fathers were colorblind. When I say that male has normal vision, that means the male will be actually normal. The male will be actually normal. When I say female has a normal vision, I have to consider one very important thing. She can have normal vision if at all she is like this, or probably when she is like this, when she is a carrier also, she will have normal vision only. So I'm not sure whether she is this or this. How will I make sure? Basically, if her parents, Baba, if her parents basically had color blindness, she will have the gene for color blindness, because from either of the parents you get one X chromosome that can be color blind, right? So here it is written, though their fathers were color blind. So obviously her father was color blind, and from the father you get only one chromosome, that is the X chromosome in the case of females. So this chromosome has come, which is why over here she is definitely going to be a carrier. We have got an understanding of that. So normal vision, always consider when you talk about females, you have to consider for the next thing, that is whether they are carriers or not, even though it might be written they are normal. Okay, now the probability of their daughter becoming colorblind is, if at all you're considering this and you're talking about their daughter becoming colorblind, let's calculate this and this goes, you get X and X. She is a normal female, right? Now this and this goes, you get XC. What is going to happen? Over here she is going to be a carrier, but again she will be having normal vision only. Okay, now yes, over here X and Y, normal male, it's a normal guy out there, and finally you have what? XC and Y. This will be what? A color blind son. This will be a colorblind son. You have been asked about the probability of their daughter becoming colorblind. So in that case, the answer for this question is going to be A. It is going to be A. And most of you were giving me the answer as D. Why D? You have been asked about the daughter. You have been asked about the daughter, right? Guys, you have been tricked. You have not been asked about the son, you have been asked about the daughter, and you have been asked about who are going to be becoming colorblind. None of the daughters are going to be colorblind, right? It will be 0. Yes, daughter will be colorblind only when the father is affected and the mother will be a carrier, or if she has a disease. Okay? Even the father is carrier. How can a father be a carrier? Father can never be a carrier. Father will have the disease if he has it. It's clearly written, both male and female have normal vision, though their fathers were colorblind. Okay, and when you say that male is having a normal vision, he cannot be a carrier, he can just be like this. You get it? Yes, Rathod, you are getting it in the WhatsApp, so make sure you join it. I answered it as A. Mamta, awesome. Swapna, also great. Did you get it? Guys, okay. Makes sense. For those of you who have still not got it, listen to me. Male and female have normal vision. This has been written. For a male to have normal vision, he can never be something like this. He will be colorblind. There is no option for him. Okay, he will be having normal, then it will be like this only. For a female to have normal vision, Baba, she can be like this, normal, or she can be like this also. Why? Because there is one X chromosome protecting her, overshadowing the effect of that. So you have to look for the females. She can be a carrier in this particular case. Baslingappa, why not C? Have you understood it? Why not C? Because over here, whenever you cross this female is not affected, this female is just a carrier. 2 females are being born and 2 of them are not being affected, and here we are asked probability of daughter becoming colorblind is. So 0%. Clear? If it's clear, give me a thumbs up. If you still have a doubt, please ask me. I'm here for you. Okay, I am here for you. I'm here for helping you out. Why not D? How can it be D? How can it be 50% of? Do you see the females are colorblind over here? No, none of them are. So how can you say that 50% of them are going to be colorblind? One is a carrier, one is normal, right? Please explain once again. Okay, I can explain. Okay, let's go from the basics. Let's go from the basics. Listen, this is important, and color blindness related questions have been repeated, which is why I'm telling you. Please understand it. Okay, both male and female have normal vision. Okay, let's decode it. When you say male have normal vision, he will be always like this. He cannot be like this because there is no male carriers in color blindness. If he is like this, he has the disease. He cannot be having normal vision. So this option is not there. Okay, when you say female is having normal vision, you have to consider this option. Either she can be like this, normal vision, or probably she can also be like this. She can be a carrier, but she'll still have normal vision. Why? Because there's a normal X chromosome to overshadow the effect. Now, how will I be sure that whether it's this or this? Let's see. I have to talk about the parents then. I have to look for the parents. So here it is written, both male and female have normal vision, though their fathers were color blind. Now, when I say a father is color blind, that means his chromosome is like this. One X chromosome has been affected, and the fathers, Baba, what do they pass to the daughters? Only the X chromosome. So obviously this X chromosome is going to come to the daughter, right? This affected X chromosome is going to come to the daughter, and that has come. That will be there. That will be there for sure. How many daughters ever he make, he will be carrying this X chromosome only, right? And that particular thing is going to go like that. And when I say she has to be normal, that means she has a normal X chromosome as well. Done. Now the probability of their daughter becoming colorblind is that the question, right? So we have to look for the daughters only. Be specified for the daughters. When you do this with this, you get XX. Is she a daughter? Yes. Is she normal? She is normal. So I'm just writing she is normal. Okay. When you do this with this, you get XXC. Is she normal? Yes. Why? Because there's a normal chromosome out there. So I'll write she is normal, but always remember she is what? A carrier. She is a carrier. She's not having the disease. Two females are there, none of them are being diseased, and that was the question. Probability of the daughter becoming colorblind, it will be 0%. Now let's look for the sons. When I look for the sons, this and this comes together, XY, he is going to be normal. When this and this comes together, it's going to be XCY. Over here, if at all you see, there is no other X chromosome to overshadow the effect of this, which is why this guy will be having what? Color blindness. So this will be a colorblind son. But you are not being asked about colorblind son. You have been asked about what? The daughters, and the daughters are not. So your answer for this question is going to be A. Is this clear? Is this question clear to everyone? If yes, just give me a thumbs up. Suresh, Ritika, you do not, Hina, everyone, thank you for giving me thumbs up before, and yeah, everyone for, I mean, yes, dear, I did not upload. I told you I will need some time. Obviously, actually, you know what? We should be getting the data for going ahead and making videos on college reviews. Otherwise, it will be very wrong to do so. I have told you, right? So I said I'll try my best. I'm still trying for it. Okay. Okay. Hello, Khushi. Okay, guys, so I hope this is particularly clear. Let's go ahead. The question asks for daughter, not for the son who has XUY. Yes, so it can be asked for the sons as well. Okay, it's the choice of the people who are giving you the question. It can be asked for the son, so be prepared. You need to understand the concept. Okay? Perfect. Choose the possible genotypes responsible for the lightest color of skin in human beings. What is the possible genotypes, if at all you're looking for the lightest color of skin in the case of human beings? No Syed questions are not going to be repeated, only concepts will. If son, then the answer 2 sons are being formed. Okay, 2 sons are there. So out of that, 25% of sons are being affected. Right? Yes, half. Okay. C. Venkat says C. Swapna says C. Hassan says C. Manaswini says C. Hina, Chidambar, GS World, Yash, Giri, and did I miss someone? F1. Sharon says C. RG Patil have a different answer. Okay, Yash, and okay. Guys. The answer for this question is going to be C. You are being asked about what? The lightest skin color. This is an example of what? Any idea? This is an example of what, Baba? This is an example of what? Pleiotropy, polygyny, what? This is an example of easiest question. Easiest question. Yes, tell me. Multiple alleles are. Dun-dun-dun. Okay, it can be an example of multiple allelism. Fourth exception of Mendel. Exactly, Baslingappa. Polygenic inheritance. So listen to me very carefully. Mendel said that, okay, so over here you have capital T and you have small t. Basically, these two alleles are the only ones that will be there for the coding for the height of an organism, right? But there was a change. There was an exception out there, and it is known as polygenic inheritance. It works like this. You have to be very careful while listening to me, okay? So there is capital A gene suppose, capital B gene suppose, and capital C. All of them are going to put some effect, okay, are going to show some effect in what? Your skin color. Ma'am, I didn't understand. Listen to me. What is the genotype over here? Capital A. What is genotype? Capital B. What is genotype? Capital C. If at all someone has this combination, basically they will be having the darkest skin tone out there. On the other hand, okay, let's make it lighter. Over here, let's make it small a. Let's make it small a. Rest everything is same. So what is going to be happening? The genotype is going to change. Capital A, small a. Capital B, capital B, capital C, capital C. Is this going to be lighter than this one? Definitely yes. And if you want the lightest of them, what do you have to do? You have to make sure that each of these alleles are going to be what? Each of these alleles are going to be recessive alleles. Now the combination will be what? Small a, small b, small b, small c, small c. Over here you will get the lightest skin tone, and since multiple genes, this gene, this gene, this gene are affecting for one particular character, I'll say it is polygenic inheritance. It is an example of what? Polygenic. Multiple genes are being affecting one particular trait or one particular character out there. That is why polygenic inheritance. This is found in two cases, in the skin color and also in the height. For the height and the skin color, this is something that is given as an example. Wait. Okay, is this clear to everyone? And ma'am, doubt. Normal vision, ma'am. Dear, we'll solve that question, okay? And if at all, you can just wait for some time. Once all the questions have been done, we'll come back to your question, okay? Ma'am, doubt from the last question. What if the parents are 3 sons and 1 daughter? It will be 0%. Dear, you do not have to consider, as in there are possibilities of n number of sons and n number of daughters being formed. Be strict to the ratio out there. Generally, this is how four type of progenies are possible. And when you talk about 4 type of progenies, this is the condition that happens. So you have to apply it for every condition out there. Okay. Guys, is it clear to everyone? Please give me a thumbs up if at all you feel that it is super clear. Yes. How to get the PDF notes? So dear Tarun, so over here what you have to do is, you know, the live is ongoing. Click on the description section. So the first thing that is given to get the lecture notes and PDF, join the WhatsApp. So you can just click on that particular link and you will be able to join the WhatsApp. Once the class is being done, the PDF is going to be uploaded for this class. Okay? When the class is done. Ma'am, can I get the answer for the question that for skin color they will use ABC? Is it a rule? Have been discovered when they will use. Okay, so it's just ABC that you have to use. Okay? Cool? Okay. They have this obviously the separate type of genes. It's for your easiness. They have given you ABC. Cool. Next question. Question number 12. ABO blood type in man is an example of ABO blood type. Okay. Pleiotropy, incomplete dominance and codominance and multiple allelism, and select the code for the correct answer from the options given below. What do you think is the answer for this particular question? Hassan says it is C. C and D only. Okay. ABO blood type in man is an example of. Venkat says it is A. Okay. RG Patil says it is C. And RG Patil says C. Okay. Divya says D. Amma. Swapna says C. Guys, Sharan says C. Mamta says C. Mirjafir says C. Chidamba, thank you dear. Hina says C. Most of you guys are saying C, which is why I think Divya is also saying C. We should lock C. Let's see. Let's see. ABO blood type. ABO blood type means you have what? Basically, in ABO blood type, I have 3 alleles. IA, IB, IO. These are basically examples of what? Multiple allelism. You have multiple alleles, right? You have multiple alleles, right? When you have more than 2 alleles, for example, when you talk about height, you just had capital T and small t. You didn't have anything else. So it's not an example of multiple allelism. When you have more than 2 alleles out there, we say they are examples of multiple allelism. And here you have it in the case of blood group. Now the next thing that you see over here is that whenever you get a combination like this, IA and IB, when they come together, both A and B are going to express themselves. This is known as codominance. This is known as codominance. So this is an example of that also. Codominance is also being covered. Multiple allelism is also being covered. Incomplete dominance means what? Incomplete dominance means basically suppose I have IA and IO and it is not completely dominant over O. It is not completely dominant over, and which is why you see a middle character. For example, you saw that in Snapdragon. Okay, there is capital R and there is small r. You expect it to be red in color because capital R is dominant, right? But it shows incomplete dominance, which is why it will be pink, not red. You get it? So that is not seen over here. Now pleiotropy is what? Pleiotropy is different from polygeny. In polygeny, what did we see? Multiple genes coding for one trait. What if one gene is there and it is coding for multiple characters out there, only one gene and multiple characters are being affected, that is known as pleiotropy, and that is seen in the case of phenylketonuria. One enzyme is being mutated, one enzyme is not being found, which is why multiple characters are being affected in phenylketonuria. So pleiotropy is something different, not associated with this. So C and D only are the options which are correct. So your answer for this question is going to be C. Is this clear? Yes, PKU, Sagar Chopra. Perfect. Is this clear? Very easy question, right? Can you give me a thumbs up if at all it is clear? P for pleiotropy, P for phenylketonuria. Exactly. Don't go P for polygeny and P for phenylketonuria, okay? That is why I say shortcuts are important, but you have to remember certain things. Zuhal says, ma'am, can you say the difference between multiple allelism and polygenic inheritance? Polygenic, okay, in polygenic inheritance, it means that, suppose, over here, I am considering this, right? I am saying that multiple genes are coming together to show me an effect on one particular character, right? Right. Only one particular character out there. This multiple genes affecting one particular character is polygenic inheritance. You could have this, or probably in this particular case, you have capital T and small t. You do not have multiple genes. You just have these genes for coding for what? Height, according to Mendel. Only one set of gene. But here you have multiple set of genes coding for one particular character. That is why it is polygenic inheritance. Now, multiple allelism basically means that for one particular character, okay, for one particular character, you have multiple alleles. So here you saw there is capital A and small a. There is nothing in between. Okay, there is nothing extra there. Only two alleles are there of this particular gene. When you talk about blood group, okay, when you talk about blood group, what is going to happen? In the case of blood group, you have IA, this is one allele, IB, this is another allele, IO, this is another allele. Three different alleles are there for one particular gene. This is one gene which is having three different types, three different varieties of one particular gene. Okay, that is why it is known as multiple allelism. Polygenic inheritance means over here, multiple set of genes are coding for one particular trait. Here, one gene have multiple varieties. That is the difference. I hope it is somehow clear. Is it clear? Multiple set of genes. This is only one set of gene. This gene has different types of alleles out there. Okay, there are different varieties of this particular gene. Here in this particular case, you do not see that. Different varieties is not being seen. Is it clear? Drinks break for only 30 seconds. Okay. Hi, Miss. Hello, Pujari. Buslingappa says clear. Okay, thank you so much, guys. Explain once again. Missed this one. Pooja, are you talking about this one? This question? Clear, children? Clear, clear, clear? Yes. Okay, fine. You need an explanation, I'll tell you. ABO blood group is an example of, is being asked. ABO blood group is an example of multiple allelism. Why? Because over here, in the case of this particular gene, suppose there is a gene over here. This gene is for the blood group. Here, you do not have just two options. Basically, you can have IA or IB, or basically you can have IO also. These are what? Multiple alleles. This can come in different combination. Sometimes it can be IA, IO, it can be IA, IA, it can be IB, IO, it can be anything. But how many alleles do you have over here? Three different types of alleles are there for blood grouping, which is why it is an example of multiple allelism. Now, codominance, why? Codominance because suppose I have from here IA, from here I have IB. When I have a combination like this, basically, the dominant one should express itself. What is happening over here? Both of them are expressing themselves, which is why we have a blood group AB, which is why we have a blood group AB. This is known as codominance. So, which is why C and D would be the example, or basically the answer for this particular question. Is this clear now, Pooja Pujari? And Syed also? Okay, why not incomplete dominance? Because incomplete dominance means it's not completely dominant. Here both of them are equally dominant, codominance. Okay, and when you talk about other scenario, that is IAIO or probably IBIO, they're completely dominant over O blood group. There is no incomplete dominance. Incomplete dominance is seen in the case of snapdragon, that is Antirrhinum majus. There you saw that capital R is for red, okay, and small r is for white. And when you expect a combination like this to come up, you expect that basically it will be completely dominant and it will give a red color. But over here it is not giving red color, it gives pink color. That means it is what? Incompletely dominant out there. So incomplete dominance is seen in snapdragon. Okay, it's not a character over here. Pleiotropy. Pleiotropy means one gene having multiple effects. So here this gene do not have multiple effects, they are only coding for the sugars on the surface of the blood cells. They do not have any other multiple effects out there, which is why I'll say that this is wrong as well. Is this clear now? You are most welcome, children. Yes, cool. Okay, Sagar, when we reach 15th question, I will give you a break. Okay, when we reach 15th question, I think I'll be giving you a break. Clear? I'll give you 5 minutes break, not 30 seconds. Okay. Example for autosomal hyperaneuploidy is autosomal hyperaneuploidy, Down syndrome, Klinefelter syndrome, Turner syndrome, and hemophilia. What do you think is the answer for this? D. Who has a blood group? Dragon. Other than this, only dragons, not us. Okay, Rajji Patil says D is the answer for this question. Hassan says 13A is the answer. Okay. And Mamta says A. Rajji Patil still says D. Guys, have you read the question? Have you read the question? Even if you're someone who does not understand what do you call aneuploidy and things like that, at least you know the term autosomal. At least you know the term autosomal. Autosomal chromosomes means the chromosome apart from the sex chromosome out there. Now, which is the one where the sex chromosomes are not being harmed? Just think like that. If you have no idea about hyperaneuploidy, at least you have an idea about autosomal, which is why you should be able to give the answer for this. Children, do not randomly say it is D. Why will it be D? Hemophilia, Baba, hemophilia is X-linked recessive disorder. It's X chromosome linked, sex chromosome linked. How can that be autosomal? A is autosomal, no Baba. A will be autosomal. Down syndrome is autosomal disorder. Dun, 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 dun. All my dear children, understand, we have to be careful while reading the question. The question might seem very scary, or it might feel very new to you, but there might be some terms in there which might be, what do you call, an older thing for you guys. Answer for this question is going to be Down syndrome. Okay, hyperaneuploidy means basically one extra chromosome. That is what you mean over there. And it is autosomal, so it is definitely Down syndrome, because here 21st chromosome is autosome and it has an extra copy. Klinefelter syndrome is basically sex chromosome is extra. In Turner syndrome, X chromosome is not there. In hemophilia, again X chromosome is affected, and they're not even autosomal, they're all allosomal. Got it? Sanjana ma'am, these questions will help for NEET also. Highly doubtful, but yes, in some ways. But yes, for NEET questions, you have to practice more of NEET PYQs itself. These can be helpful for sure because concepts are being done. But again, the difficulty level might be going up a little more. Okay, clear? Exactly, everyone. Hassan, B, C, D, and are allosomal. Can you differentiate between autosomal hypoploidy and hypoaneuploidy? Ayyo. Hyper as in basically you are gaining a chromosome. Hypo as in basically you're losing a chromosome. There is no specific term like that even in your NCERT. Just telling you. Okay? Yes. Akshat, ma'am, good morning. I have purchased the Comet Key course, but I do not know where to access. Go to Diksha Vedantu's app, and that might be sent to your WhatsApp or email. You have to go to the Diksha Vedantu app and then log in. Okay? Okay. Guys, let's go ahead. Question number 14. A human male. From here, so these are the questions which can go like zoom through your brain. A human male is heterozygous for autosomal genes A and B. He is also hemizygous for hemophilic gene B. What percentage of sperm will carry ABB genotype? Come on, let me see. Fatima says the answer for this question is B. Hello Fatima, by the way. Okay, what about others? What about others? D. RJ Patil says D. Okay. Mir Jafar says B. Chidambar says B. Okay. Okay. So, Sanjana had a different option. I don't know. Okay, so that's a good option. Was hemizygous. Okay, so reason. Okay, I'll tell you. Most of you have given me the wrong answer. Now listen to me very carefully. Only one person I see have given me the correct answer. Now, I'll tell you what is hemizygous and then you have to tell me the answer for this. Okay? Hemizygous means basically they do not have the copy of the allele. That is, when you're talking about a male, right? And when I say it's hemizygous, that means probably the allele is over here, but it does not have an alternative allele or basically a companion allele on the next chromosome. Why? Because it's a Y chromosome. So basically it does not have a companion or opposite allele out there. Whenever I say that is something like this, I will not say it's hemizygous because it has a companion over here. Okay, you got it? But here in this particular case, there is no B gene on the other side, or there is no B allele on the other side, which is why we call this condition as what? Hemizygous condition. This is known as hemizygous condition. Got it? Now you gave me the answer for this. Hemizygous means this is a thing, and I am saying that male is heterozygous for autosomal gene A and B. When I say they are heterozygous, that means it is going to be like this. If I say that they are homozygous, it would have been like this. But now here they are heterozygous. Similarly, for capital B also they are like this. You have been asked what percentage of sperms will carry ABB genotype. What percentage of them will be carrying ABB genotype? Now, any answer? I'll give one more clue. Basically, this can come in different combinations. They can come from here, capital B can come from here, capital A can come from here, XB can come, anything can come. So basically, if you're arranging like that, just give me a second. I'll make the arrangement so that it will be easy for you guys. It will be something like this. Okay, so basically you can choose from here one, from here one, from here one. Similarly, here one, here one, here one. Whatever combinations you make, then you have to tell me the answer for this. Hina, okay. Ma'am, explain once again for hemizygous. Hemizygous means, Baba, it does not have an alternative allele on the other chromosome. So usually we have no. So when we say capital T, we expect that other gene will be there over here. Either it can be capital T or it can be small t. I do not know. Either of them. But over here it does not have a companion allele on the other side. So when I say it's like this, I will say it is not hemizygous because it has a companion allele out there. But here it's hemizygous condition where it does not have a companion allele because it's a Y chromosome on the other side. Now you give me the answer for this question. What will you think? A. Okay, let me see. Divya Prajapati says D. Hazira says D. Okay. The answer for this question is going to be D. It is not A, B, C, D. It is going to be D. Listen to me very carefully. From here, suppose small a have come. From here, suppose small b have come. From here, you have either an option for what? XB, right? Or probably you have an option for Y to come. Do you ever see something like ABB? No. It has to be XB, but here it is mentioned as ABB. That is not possible. ABB is not possible. ABXB is possible. Got it? From here you can get only from this, right? You do not have any other choice out there. Either this combination or this combination. The thing that they have written is ABB. That is not at all possible, which is why your answer for this question is going to be D. Is it clear? Miss, what is your next class time? Pooja Pujari, have you liked this class? Okay, anyways. So my next class time will be on, I think, Monday. Afan, dear, it will be on tomorrow morning, okay? RJ Patil, yes, you were the first person to answer it. I clearly saw that. Clear? Ma'am, NEET ke idella use agata. Listen to me carefully. So in NEET, obviously the question level is a bit more higher. Definitely it can help you, but you have to practice NEET questions separately. Okay? Cool? Ma'am asked the percentage of sperm will carry ABB is 50%. 50. No, Baba. It's not 50. It is going to be 0% only because of the combination like this. If at all they had mentioned, okay, if at all they had mentioned A, B, X, B, and then you are asked this question, then it will be 50%. Why? Because 50% will carry this, 50% will be carrying this. But they have mentioned ulta. They have not mentioned A, B, X, B. Instead, they have mentioned A, B, B. What is that? What do you mean by this particular B? This B and this B is the same, right? So that is why the answer for this is that. Clear? Sunday holiday, right? Yeah, it is holiday for us, but we are taking classes for you guys. Even today, this Saturday should have been a holiday. It's a second or fourth Saturday, something like that. But we did not want you guys to miss on anything, and it's a 15 days challenge. We have to be very, very, attentive over here. We have to give our best over here, right? So that's why we are coming. Cool? Yes. Next question, is this particular question clear? 15th question is super clear, I hope, right? By the way, is there anyone who feels that it was not clear? Sharon, when will be the physics class? Tomorrow. At what time? I do not know. I have to confirm because it's Sunday. Yeah, anyways, it is there tomorrow, okay? In the afternoon time, it will be there. In the morning, you would be having chemistry. In the afternoon, you would be having that. Okay, hello Preetham. Hello. Okay, Fatima says the answer for this question is B. Okay, RJ Patil says A. Hassan says C. Venkat says A. Venkat says A. Okay, Mamta says A. Swapna says C. Manaswini says A. Shifan is there. Hello. So most of you guys are saying it is A, and some of you are saying it is B. Okay, which is why you have to look forward for it. Test cross in a pea plant is a cross between F2 tall plant and a recessive plant. Okay, I'll explain what is test cross and then you tell me the answer for this. Okay, suppose I have a condition like this. The plant appears to be, the plant which is there, it appears to be tall to me. But tall can be like this, homozygous condition. Or tall can be like this, whether this is dominant, heterozygous condition. How will I understand whether it's tall like this or this? Basically, I want this because this is pure breed. How will I get to know that? I only have one option, to cross it with a recessive parent. When I cross it with a recessive parent, that means you're talking about small t. Here also, you're talking about small t. I'll get my answers. Why not dominant parent? Because if you're crossing anything with dominant thing, only dominant character will appear. No. We do not want that to be seen. That is why we cross it with recessive parent. Okay. Now, when you do that, let's see what are you going to get. So, small t over here, capital T over here. When you cross this with this, capital T, small t. Capital T, small t. Ayo, where did it go? Poya. Lampoch. Yes, got it back. Capital T, small t, and then finally capital T, small t. All of them, if at all you see, they're capital T, small t. All of them are going to be tall, Baba. Right? So I say all of them are tall. Let's see if the answer is different over there. Okay? When I cross this with this, small t, and here you get capital T, small t. When you cross this with this, capital T, small t. When you cross this with this, capital T, small t. When you cross this with this, small t. When you cross this with this, small t. Abba, here two of them are going to be tall and two of them are going to be dwarf. Right? Two of them tall and two of them dwarf. With that, what I understand, that basically the plant which was appearing tall to me probably could have been like this. If at all I get something like this. Okay, on the other hand, if at all of them are tall, I understand that plant which is tall is actually homozygous tall. This is test cross, where what we are doing, we are going back. Okay, we are going back to the recessive parent and we are crossing them. So a cross between F2 tall plant and recessive parent, a cross between F2 dwarf plant and recessive parent. Why will you do that? A cross between F2 tall plant with a dominant parent, you cannot do that because only dominant traits will appear. And then over here, a cross between two F1 plants. Your answer for this question is going to be A. Got it? Arey Baba, yo. Got it? Yes, this is back cross. This is an example of back. Listen to me. All test cross which is there is a back cross. You go back is an example of back cross only. Okay, not all back crosses are test cross. Back cross means when you're going back and crossing with any of the parents. It can be dwarf parent, it can be recessive parent, it can be dominant parent. That is back cross. When you're specifically crossing with a recessive parent, you call this as a test cross. Clear? Ma'am, should we write KCET for health allied sciences? I have to check. In the 5-minute break, I'll check and tell you. Okay? I forgot I made the video yesterday only. Okay. First PU syllabus. First PU syllabus, everything which is there in the 11th grade. Yes. One short video is useful. Definitely it is going to be useful. Okay, guys. Let's go on a break because some people some among you are demanding for break since a very long time now. 5 minutes break work, right? Everyone, 5 minutes break work, right? So, what is the time now? It is 5 minutes. Cat break. Go drink some water, have some nice time, and be right back. I'm giving 1 minute extra. Bye. 10:55. Oh my god, this looks monstrous, but yes, this is me smiling. Okay. I don't want more than 30 seconds. Guys, we'll take 5 minutes break. Even I will go just drink some water and search for health, allied sciences and come back, okay? Round figure 11. Popo. 10:55 will come. Bye, 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 bye. Natil Evadiya, ma'am? Malayali. Shama. Natil Wayanad. Okay, bye, 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 bye. Hello guys, you know what you saved me. Okay, so you have got the most, what do you call, creative teacher out there with that. Is it clear? Am I audible? I'm so sorry for being 1 minute late because you know what, I actually forgot. So someone asked me a question about health sciences. I was just reading that, and yes, you do not have to give allied health sciences courses. You do not have to give a CET examination, but you have to register for it. I hope you had, and you just have the ranking system will be based on your PU2 marks. Okay. So Sham, one very important thing, your ma'am, even though she is a Malayali, she does not know to read Malayalam so well, okay, because she has been raised up somewhere else in North India. So, but yes, I think so Kadicho is what I was able to read. Amme also, and between that, what was that I was not able to read it. So I'm so sorry, but yes. Okay, so I hope we are back. Okay, everyone is back. So let's go ahead with the next question out there. Central India, exactly. I was born in Indore, Madhya Pradesh. Yes, exactly. Hello, Kit Kat ma'am. Hello, hello. So I've got multiple names now. Let's go ahead with the next question then. 16th question, and now from this point onwards, we have to be a bit fast. Yeah, we will be a bit fast, and also we will be too much productive. Please share this video with your friends. Let's go ahead with the next question. Find the incorrect statement among the following. In sex-linked recessive traits, the gene is transmitted from unaffected carrier female to some of the male progeny. In sex-linked recessive traits, the gene is transmitted from unaffected carrier to some of the male progenies out there. You have to find what is incorrect. Accumulation of phenylpyroic acid in the brain results in mental retardation. Does it? Individuals affected by Down syndrome will have congenital heart disease and more intelligent. Is it? Turner syndrome is caused due to absence of one X chromosome. Is it? What do you think is the answer for this? Welcome back, Macha. Welcome back. RG Patil says the answer for this is C. Hassan says it is C. And Shifa says it is C. Hina Kausal also says, Chidambar says C. Preetam says C. Halesh C. Mir Jafar and Sanjana, Vijay, Mallika. Hello, Mallikarjun, FR Pan, everyone. Okay, ma'am, what about practical based questions? We will be solving them separately, okay? Yes. And yes, so Vijay and Vishwanand and Sagar, RS, oh my god. Everyone is saying the answer for this question is C, which is why I have to look it up. Is it C? Why do you think it is C? Because listen to me very carefully. We'll go one by one, right? So it goes like this. In sex-linked recessive traits, for example, when you're talking about hemophilia, suppose, and you are saying that the female is what? Unaffected carrier female. So she is a female who is unaffected, but she is a carrier. Do you think that she can pass it to the sons? Obviously yes. To the sons, the female has to give the X chromosome. It can be this X chromosome which is affected, it can be this X chromosome which is not affected. Either way, but they can pass it. So this is correct. Now accumulation of phenylpyroic acid in the brain can lead to mental retardation. This is correct, and this is seen in phenylketonuria as well. Correct. Now individuals affected by, okay, just a second. Yes, individuals affected by Down syndrome will have congenital heart defect, correct. But are they more intelligent? No, Baba, you see mental retardation out there, which is why this particular thing makes the statement wrong. Turner syndrome is caused due to absence of one X chromosome. Yes, monosomy is what you call it. One X chromosome is lost, so rudimentary ovaries and everything will be there. Your only answer for this question is going to be C. And my dear children, are super correct. Okay, I think every one of you gave me the correct answer. Super awesome. Super proud of you. If it's clear, can we go ahead with the second question? Just give me a thumbs up. If it's clear, can we go ahead with the next question out there? Chidambar, why are you sad, da? You also gave me the correct answer only. Okay. Clear. Perfect. Thank you so much. Let's go ahead. A colorblind man, again colorblindness thing, okay, 2025, the previous year question, this is very recent. A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be a colorblind man, that means over here X chromosome is affected, marries a carrier woman, that means she just has a one chromosome being affected. The percentage of their colorblind progeny in the next generation will be what? And you guys are saying me the answer for this question is what? Okay, thank you. Okay. Hina Kausar, everyone, okay. RJ Patil says B, Shifan says B, Swapna says A, and Sham says 50%, okay. 50% means what? B, okay. RJ Patil says B, Rathod says B, most of you guys are saying B, Vishwanant also says B, and GS World says B. Anish is there. Hello, Anish. Preetham, Nasreen, Vijay, Manaswini has a different answer. Okay, guys. Sagar also said B. Kiran, B. Okay, guys, listen to me carefully. Over here, we have to look for the colorblind progeny. Male or female is not been mentioned. So let's try to fuse it and see. This and this comes, you get a carrier female. She is what? A carrier female, but she is not going to be colorblind. So no. Okay, so whenever it's not colorblind, what do? You write it and then you put a cross also, so that you get to understand easily. This and this, basically both X chromosomes are affected. That means it will be colorblind. Put a tick. Okay. Now this and this XY chromosome, very normal guy, so cross. This and this, over here one X chromosome is affected, but that's enough for a male to be colorblind. So basically I'll say this is also colorblind. Out of four of them, 2 of them are affected. That means half of them are being affected, are being colorblind, one female and one male. Can I say half is basically what? 50%. Answer is going to be B. Clear? Is it clear? This is 2025 question. If at all you were able to do it, that means that you have understood the question or the concept really well. Done? If it's clear, just give me a quick thumbs up. Everyone, come on. And this is how we learn. If you have made a mistake over here, understand, rectify that mistake, do not do it again. Understand your mistake, okay? Let's go ahead. Perfect. In the following symbols used in pedigree analysis, identify the symbol that denotes consanguineous mating. Consanguineous means mating between the parents. Sorry, mating between the relatives, not the parents, mating between the relatives. That is what you mean by consanguineous mating out there. So how do you denote that? It's a very simple question, easy answer. A. Preetham says 18B, okay. Shifan says B, Manaswini says B, Hina Kausar, everyone. Tasu is also there, hello. Anish, Hassan, Mohammed, Manaswini, Vijay, and GS World, Sham. Okay. Everyone are saying B, so basically the answer for this question is actually B. How do you remember it? Always remember, when you have got a square and you got a circle and you have two lines in between, that means the mating is stronger. It has to be between relatives. So obviously this is going to be correct itself. Okay, same question came last year, is it? Okay. Next, from the following pedigree chart of a family, what do you think is this type of trait that is being shown over here? Okay, it is an autosomal dominant trait, it is an autosomal recessive trait, it is an allosomal dominant trait, and it is an allosomal recessive trait. What do you think is the answer for this particular question? Ma'am, board exam bandi, yes, exactly, it came. The previous question, right? Okay. B, 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 B. Some people are saying B, some people are saying D, C, and some people are saying D also. A, B, C, D option for me. This means that our pedigree analysis part is not that strong, right? But most of you guys are saying it's B. It's B is what? Autosomal recessive. First of all, listen. Cancel out the option that does not make any sense over here. Is it dominant disorder? No. How do you understand that, ma'am? Basically, if it was dominant disorder, it will be found in every single generation. Either one of them is going to be diseased in every single generation. But over here, you see that these guys are not diseased, but their children are. You get it? So that means there is skipping of generation. And if at all you feel that there is skipping of generation, it has to be recessive disorder, not dominant. Dominant one dominates in every single generation. So dominant one, I am going to cut it off. What are we left with? We are left with these two options only, that is B and D. Now when you talk about B and D, one is autosomal and one is allosomal. Allosomal means sex-linked. How do we identify that? First scenario that is there, usually in sex-linked traits, the males are the ones which are usually more affected. The males are the ones which are going to be usually more affected. Here when you see this generation, male and female equally affected. Here female is the one that is being affected. That simply shows us it cannot be what? It cannot be autosomal, it cannot be allosomal, that is sex-linked. Probably it can be autosomal, which is why your answer would be this. Which is why your answer would be this. And always remember, if this also you are not able to understand, I'll give you one more thing. Let's sum it up. First of all, dominant disorders do not skip the generation. Recessive disorders, they skip the generation. And here skipping is being seen. That is why it is recessive. Now it is autosomal or allosomal. Autosomal means both the sexes could be equally affected. In allosomal, it is going to be the males who are going to be mostly affected. In the first generation, you see male and female are equally affected. In the second generation, you see that female is being affected. But still we are not sure. Okay, still we are not sure. Understand, in sex-linked disorders, you will see crisscross inheritance. That is, from the female, you will see males are being affected. From the males, females are being affected. But that has not been seen over here. From the females, female is only being affected. There is no crisscross inheritance pattern that is being seen over here. That is why, again, it is not going to be sex-linked. It is going to be autosomal only. Is it clear? In KCET, they will ask easy question based on pedigree chart. Definitely, yes. They will be mostly asking easy questions, but you should know the basics, yeah? Clear? If it's clear, just give me a quick thumbs up. If it's clear, just give me a quick thumbs up and then we'll go ahead with the next question. Thank you so much, children. Okay, perfect. Lots of love. Macha, how many questions are there? Macha, how many questions are there? I think in total I have taken 50 questions. Okay? Yes, 50 or 45? I don't remember. Okay. Anyways, the gene disorder phenylketonuria is an example for the gene disorder phenylketonuria. Thank you so much, guys, for everyone who gave me a thumbs up. Okay. Now, yes, the gene disorder phenylketonuria is an example of polygenic inheritance, pleiotropy, multiple allelism, or multiple factor. What do you think is the answer for this question? B, 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 B. Most of you guys are saying B. Okay, Hassan, RJ Patil, and Swapna, and Sanjana, and Tasu Chidambar. Okay, Manish, Shifan, Malikarjun, Hassan. Okay, Sanjana, RJ, okay, Vijay, everyone. B, 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 B. B is what? Pleiotropy. Yes, this is an example of pleiotropy. It cannot be example of polygyny. Polygyny means multiple genes affecting one particular character. For that, skin color and your height comes to be the example. Pleiotropy means one gene is affected. Because of this gene, multiple characters are being affected. And I can explain you with this. So the gene that is there is the gene for phenyl hydroxylase. This particular gene, okay, the one that codes for this particular enzyme is being affected. So when this enzyme is not there, what are the multiple effects that is going to be shown? Let's see. First thing, phenylalanine, which is there, it's an amino acid in our body. It is not going to be converted to tyrosine because this guy is not there. Okay, ma'am, it's not getting converted. What's the problem? First problem is that this guy is not getting converted, which is why it will accumulate in the form of phenylpyruvic acid. And when it accumulates in the form of phenylpyruvic acid, Baba, you have a reason to basically lead to what? Mental abnormalities or mental retardation as well. That is the first problem, okay? The first character that is being affected. The second character, when tyrosine is not made, okay, the tyrosine is not made, there is a very important skin pigment which is also not going to be made because this is super important for the formation of the skin pigment. And if this skin pigment is not being made, or basically coloration pigment is not being made, what is going to happen? You will see hypopigmentation. You will see hypopigmentation, that is your hair, your skin, everything will be having a reduced pigmentation out there. The color would be changing up. That is known as hypopigmentation, or in some cases they have also written as reduced pigmentation. This is the second character that is being affected, which is why we say it's an example of pleiotropy. One gene being affected, two characters, or basically more than two characters been affected for that. P for pleiotropy and P for phenylketonuria hazotic. Pleiotropy is a small word with penalty area in a long word. Okay. Okay, so that's for you guys. That trick is for you guys. Fifth exception of Mendel. Exactly, Baslingappa. I hope this is clear, guys. If it's clear, can you give me a quick thumbs up? Everyone, if you feel that it's clear. Ma'am, PKU chromosome disorder. Yes, it is an autosomal chromosome disorder. Okay, it's not sex chromosome. It is an autosomal recessive disorder that is there. So it's not on sex chromosome. The gene for phenylketonuria, or basically phenylalanine hydroxylase, is found on the autosomes, and this gene is being affected, is being deleted, which is why you have a problem. Okay. Thank you for giving me a thumbs up. Lots of love. Let's go ahead. The next question. A sickle cell carrier marries. Again, be careful. A sickle cell carrier man marries a sickle cell carrier woman. What is the probability of their children being born genotypically normal and sickle cell anemic respectively? This is not a previous year question. This is a new question. From here, we start with new question. I think 5 of them are there, which are new. 21, C is the answer. Chidambar says B. Vijay says B. Muhammad says A. Rathod says B. So I see A, B, C options are there. One more option you give me, then it will be A, B, C, D. Most of you guys, I see it's, yeah, you're giving me the answer as B only. So we have to analyze. Sickle cell carrier, whether it be a man or a female, whatever it's going to be the same thing only. That means they will have one gene being affected. I cannot write it over here. One gene is going to be affected. So let's do that. They are carrier, that means they have an affected gene over here. That is denoted by this. Now when you cross it, let's cross it. This and this comes together. I get HBA, HBA. What is it, Baba? It is going to be normal. Make a tick mark. Okay. This and this, you get HBA and HBS. What is this? This is going to be a carrier. This is going to be a carrier. What are we find? We have to find genotypically normal and sickle cell anemic respectively. How many of them are genotypically normal and sickle cell anemic? Okay, so this is also carrier. So again, it's found to be normal itself, if at all we want to say like that. Now, if you cross this with this, HBA, HBS. Again, this is going to be a carrier. Okay. Now, finally, HBS, HBS. This is going to be affected. This is going to be sickle cell anemic for sure. Right? Right. Now, what is the question that you have over here? Genotypically normal. Genotypically normal means they should be having a condition like this. And sickle cell anemic means they should be having a condition like this. They are also normal, but genotypically they are not. They are going to be carriers out there. Right? So can I say out of 4 of them, 1 is going to be affected? So basically that means if 1 by 4 would be what? 25%. One of them is going to be sickle cell anemic. Similarly, 1 by 4 over here is also going to be normal. So again 25%. So your answer for this question is going to be what? Your answer for this question is going to be what? Is it clear? Is going to be C, not B. It is not going to be A, it's not going to be D, it is going to be C. Is it clear? Why have you not considered carriers over here even though they are normal? Because here a term is written, genotypically normal. That means the genotype also need to have the normal alleles. That is why you cannot consider them. You cannot consider them. Dinka chika, cool macha, clear. Perfect. Okay, important. I hope it's clear, and with that hope, I'm going ahead to the next question out there. Question number 22. Oh, yes, Shantanu. Shantau. Okay? I've changed everyone's name. Guys, please do not mind doing that. I'm so sorry if I'm doing it, okay? Okay, perfect. Okay, keep get clear. Question number 22. How many chromosomes are present in each cell of a person suffering from Down syndrome? In each cell of a person who is suffering from... Ma'am, actually I guessed it correct. Is it, Sanjana? Okay. But anyway, I hope you got the concept right. Yes. D. RJ Patil says D and Tasu says D. Swapna says D. Leelavati is there. Hello dear. Says D. Okay. RS says 46. Vishwanand and Ravi Shankar, Mallikarjun and Shanta and Nikhil. Hello. Everyone. Says D is the answer. Yes, the answer for this is D. Why it is D? Listen, in Down syndrome, you would be having, this is a 21st chromosome, suppose, a pair of 21st chromosome. You have an extra chromosome out there. This is going to be the extra one. So usually in our body, how many chromosomes do we have? Autosomes. 44 autosomes are there, and then we have the sex chromosomes, XY or XX. So this is 44 chromosome, this is 45th, and this is 46th chromosome. General condition, we have 46 chromosomes. Here, autosome is extra, so instead of 44, I will be writing 45. And then you can have XX or basically XY. But 45, 46 and 47. 47 number of chromosomes are going to be there in general nature, which is why D is going to be the answer. And everyone who is there who gave me the answer, Surya, hi, everyone, always Sheikh, hello, Muhammad, Hina, everyone, perfect. And everyone who gave me the answer before also I read out your names, you are doing awesome. Sagar, everyone, cool. If it's clear, can you give me a thumbs up, guys? Clear, thumbs up needed, fast. Thank you. Okay, perfect. Question number 23. Sperms in case of drones are produced by. Sperms in case of drones. Thank you guys for the thumbs up as well. To you too, Nikhil. Okay. So sperms in the case of drones are produced by, what do you mean by drones? Ma'am, drone, camera drone. No, you're talking about, arre baba, come back. Drones as in, Chidamba says A, Tasu says B, RS says B, okay, RJ Patil says C, A, B, C. Most of you are giving me options from A, B, C. What are drones? First of all, the male honeybees are known as drones. Okay, the male honeybees, they have a specific name, you call them as drones. You know, in honeybee there is very social organization out there. Some females are going to be workers, okay, and only one female is going to be the queen, and the males who are there, they are just for the purpose of mating. That is why you call them drones. How are males being formed in the case of honeybees? You know that females have diploid number of chromosome, that is 32 number of chromosomes, and males basically they have haploid number of chromosome, that is only 16. So when they have to form the gametes, okay, they cannot undergo meiosis because again it will be half. So they undergo mitosis. They will be undergoing mitosis to form the gametes having 16 number of chromosomes each. The females will undergo meiosis. It's a reductional division so that it becomes half to 16. So sperm in the case of drones are produced by mitosis. The answer for this question is going to be B. So all of you who said A as the answer would be wrong. Guys, do you know haploid-diploid sex determination? If at all you know about that, then think about it. Exactly. Whenever you get a question which is out of the box, which you never remember at all, please try to go back and think for the concept and try to associate it with those things. Where have you seen mitosis? Where have you seen meiosis? Okay, it's in haplodiploid sex determination when we form the gametes. Those things are there. And then you try to associate with it. The answer for this, yes, parthenogenesis is going to happen later on, right? So later on, this 16 and 16 can come together to form a new female with 32 number of chromosomes, or probably the female egg can undergo parthenogenesis to form a male. Here you have been asked sperms in the case of drones. So gametes basically in the case of drones, how they are formed with the help of mitosis. Is it clear? Female have grandfather. Exactly. Females will be having father also. Males will be having grandfather. Okay. Is this clear? Is this clear? If yes, give me thumbs up. Ma'am, for this year in HSLC total marks is 525, blah, blah, blah. Sorry, I do not know about it. I am not the correct person to talk about it. So please forgive me. Okay, I can check though. Okay. Perfect. Thank you guys. Next question. Question number 24th. What will be the percentage of pure breeding F2 individuals of a monohybrid cross? What will be the percentage of pure breeding F2 individual of a monohybrid cross? What do you think? 25%, 50%, 75%, 40%. Pure breeding F2 individuals of a monohybrid cross. Pure breeding means what? Ma'am, male have grandfather. Yes, males will be having grandfather. They will not be having fathers, they will be having grandfathers. Okay. B. Okay. Tasu says B. Okay. Swapna says A. And Vishwan says B. Sagar says B. Mamta, Lavanya. Hello, Lavanya. RS, Hirmat, and Shifan, Swapna, Always, Sheikh, Rathod, GS World, Nisha, everyone. Leelavati, Mallikarjun, Shanta, Baslingappa. Okay. Hina, I don't know. Yeah, there is no question of I don't know. You have to try. Listen, what is the question? What will the percentage of pure breeding? Pure breeding means homozygous condition is being asked. F2 individuals of a monohybrid cross. So monohybrid cross, when you do it, what do you get? All of them are going to be like this in F1 generation. You have been asked about F2. So what do you do in F2? Capital T, small t, capital T, small t. When you cross them, what do you get? One is capital T. One is capital T, small t. One is capital T, small t. And the last one will be small t, small t. How many of them are going to be pure breeding? How many of them are going to be homozygous? That is a question. This is homozygous. This is homozygous. Both of them are heterozygous, right? Yes. So in that particular case, what will be your answer for this question? Out of 4 of them, 2 of them are homozygous. So 2 by 4 is equals to, wait, basically what, 1 by 2, that means 50%. 50%, half of them. Your answer is going to be what? B, Baba. Is it clear? These type of new questions, by the way, are you enjoying the type of new questions which are there? I've tried to take up some difficult questions as well. Works? Clear? Thank you guys, Sanjana, Nisha, Ravi Shankar, Mamta. Perfect. Yes, ma'am, enjoy. Okay, perfect. Now, I think this is going to be the last question. It's going to be easy for those of you who remember the dates. Yes, yes, Baba, understand. Okay, okay. Which one of the following is the period of Mendel's hybridization experiment on the garden peas? Which one is the period when he did the experiment? Try to find the answer for this. 1840 to 1, that is 1850, or 1857 to 1869, 1870 to 1877, and 1856 to 1863. What do you think is the answer for this question? Yes, ma'am, class is very interesting. Thank you so much, Arya. Okay. B. Some people are saying B, some people are saying D. Why there is a confusion between B and D, Baba? B and D. I see the point of confusion. 50-50 is written, that's why. Okay. It's 1856 to 1863. It is 1856 to 1863. For 7 years, he did his experiment, and I am pretty, I understand why there is a confusion out there. Yeah? So, yes. So, the answer for this question could be this. Good. Perfect. And please remember it always. 1856, okay, to 1863. 1. Got it? And now we come to one very important thing. That is the next chapter, but before that, one very important thing, that is, class is very excellent, thank you, Mamta, that, my dear children, we had launched KCET 30, and the classes have already started. It started from 21st of March, but if at all you feel that I am not able to follow by myself, that is, I need a structured class and things like that, I'll request you to go ahead with a crash course. It can be any crash course. We have launched KCET 30. It's a 30-day crash course. We'll get PU1 and PU2. And in biology, I have not covered every chapter of PU1 for one-shots, but PYQs of all the chapters of PU1 are being covered. For the rest of the subjects, they have covered. They will be covering everything out there, PU1 and PU2 one-shots and PYQs. And there will be question practice. There will be mock tests given to you. Everything in a structured way. It is definitely going to be helpful. Daily live doubt solving sessions are also there. If at all you want, you can take it up. The pricing for that is ₹2499, and the link will be given in the description section. Do check it out if you want, only if you want. Now there's offline course as well. There's a 3,000 rupees off going on. So yes, if you want to go ahead with that, you can check that. And ABHYAS KCET test series there, and the pricing for that is $999. And I'll tell you, it's very important. Okay, the test series, ABHYAS test series is super important. I consider you guys to take it up at whatever time. I'll request you to take it up now itself. Why? Because you can start practicing mock test. If you're someone who feels that I'm really good with my syllabus out there, you can go ahead and start giving the mock test out there. There are part tests, there is full length test. Part test means some chapters will be covered, 60 questions will be there. So look forward for that, and I'll request you, have one test series. Have a mock test series. It is definitely going to be helping you. It can be anything, but ABHYAS was something that we launched previous year and worked really well for children. We got multiple rank holders out there, and it's just available for $999. So you can consider it, right? Yes. Ma'am, when is your next class? Ma'am, when is my next class? It is going to be on Monday, I suppose. Yes, if all goes well, on Monday. Principles of inheritance and variation, game over. Exactly. And Nikhil, is that Korean or Japanese, or I do not know. I'm so sorry. I do not know the language, but yeah. Is the class finished? Definitely not yet. Okay. So let's go ahead. Are we all excited for molecular base of inheritance? And there will be 25 more questions out there. We will be completing those 25 questions, and we will be completing the live. And then, yes. Okay. Perfect. Hello, Ankita. Okay, in molecular base of inheritance, we have seen that, again, scattered way of asking questions, but still, you have got more questions from transcription, from genetic code, and also from the regulation of gene expressions. Some questions have come from this particular thing, that is replication. Some questions have come from the search of genetic material as well. But if at all you had to rank it, it will be from the structure of DNA, the structure of nucleosome and things like that, and also the other topics which are there. But anyway, we are going to cover everything is here, everything that is over here. And the first question goes like this. Make sure you read the question carefully. Find the sequence of binding of the following amino acyl tRNA complexes during translation to mRNA transcribed by a DNA segment having the base sequence that is this. Give me the answer for this. I know it might take time, but please read it through it, okay? Prajwal, hello. Okay. Hi Ranjit, hello Shanta. Guys, come on, let's do it. Side ma'am, okay, sorry. D. Okay, Ramesh was the first person to say the answer is D. Okay. Ma'am, this question is enough. It will be definitely helping you well. You have to do extra questions definitely, but yeah, this is going to create a strong base. Shifan D. Vitti says A. Okay. What about the others? RS says B. Rathod says D. Guys, you are giving me ABCD option. Macha. Actually, you know what, Prajwal? The translation does not work nicely. Amme actually means mummy. Okay. Amma means amma, mummy. Not getting, ma'am. Sanjana, you're not getting the answer? Okay, fine. I guess I have hypermetropia. Oh my god, sorry. This is the base sequence, and yeah, now you can try to arrange it if at all you want. I don't know. Listen, I don't know is never an option. This is what you have been asked a question that translation is happening. Translation happens with what? With the help of mRNA. You need an mRNA. But translation to mRNA transcribed by a DNA-based sequence of this. That means this is a DNA. First of all, you have to convert into a mRNA. Then this mRNA, you have to do the codon thing, anticodon and codon thing. How do you do that? Listen to me very carefully. So, if this is the DNA, first you have to transcribe it to the RNA. How do you do that? Basically, T will be going with T will be going with what? T goes with A goes with U, C goes with G. Why U? Because in the case of RNA, you do not have T. That is why opposite will be G. Cool. This will be 5' and the list goes on. This will be going on. Now, do I have to do the complete thing? Listen. Do not do the complete thing. Wait for your answer with the help of this only. If in the mRNA, okay, I've seen the answer. Anyways, if in the mRNA you have 5 AUG, in the anticodon loop you will be having what opposite of it? You will be having opposite of A is what? U, and U will be what? A, G will be what? C. This is going to be the first one out there. This is going to be the first one. Here in this particular option, where do you see UAC? UAC is nothing but the B1. So the starting would be B. But starting would definitely be B. Over here, I just have one option like that which starts with B, which is going to start with B. And that is B option, which is why B would be the correct answer. Got it? Did you get it? It is important. Okay? It is important. Guys, I can be fast, but there might be children who will not understand it. Okay? Got it? B machi. Perfect. So your answer for this question is this, and I hope it is clear. Is it clear? So whenever you get a question like this, first of all, transcribe it to the mRNA. If it is present that DNA is being mentioned and also T is there. T means DNA. It's not an RNA. First, transcribe it to the RNA. Make sure the direction is clearly visible. 5' to 3' you have to do. Okay, 5' to 3'. So you have to write the sequence in 5' to 3'. And then you have to go ahead with the next process out there. Once again, repeat again. Okay, I can repeat again. Listen to me very carefully. Okay? Over here, this is the DNA that is out there. You have to make the RNA. How do you make the RNA? Basically, first of all, you write 5' and then you go on putting the complementary nucleotide out there. So T is going to base pair with A. A is going to base pair with what? U. It should have base pair with T, but this is an RNA that you're making. So U. C is going to base pair with what? G. A is going to base pair with what? Again, it will be U. T is going to be A, and the list goes on. But do you have to make the whole thing again? No. Stop right there. Try to find the answer with this particular piece of information. So now you have an mRNA which has A, U, G over here. That means the tRNA which comes and attaches. These are basically tRNA, and you have to tell me what is the first tRNA that comes and attaches. That is the question. So over here, A is going to base pair with the anticodon U. U will be base pairing with an anticodon A. G will be base pairing with the anticodon what? C out there. So UAC is the first one out there, and over here you have got only B with UAC. So this is the first tRNA to come and attach out there, and over here, that is why none of the other options have B in the first part. Everyone has A, C and stuff like that. B is the one, that is the first one, which is why your answer is going to be this. Is it clear? Is this particular thing clear? If clear, give me a thumbs up fast. Okay, perfect. Thank you so much, guys. We'll move ahead with the next question. 27th question. E. coli bacteria grew in NH4Cl medium. This is 15th isotype, okay? Medium for several generations are allowed to grow in NH4Cl medium. This is N14 isotope. After 2 generations, the bacteria are isolated from the medium, and the DNA of the bacteria is centrifuged in CSCl, that is cesium chloride. The result of the density gradient of DNA is, what will be the result of this particular thing? Any idea? Any idea? So first of all, you are going to grow the E. coli in N15 medium, then you are growing in N14 medium, and then basically you are saying after 2 generation, what is going to be the case in the case or when you centrifuge the things? Fatima says it is B. Okay, Nikhil, okay guys. Maitri is there, hi, says it is D. Fatima again says it is B. What about others? Try. I'm telling you, the questions from the chapter molecular base of inheritance could be difficult but not impossible. You have to try. Keep trying, guys, not be sad. Muhammad B. Okay, Nikhil says B. Okay, Viti says B. Anish also says B. Okay, what is B? Both hybrid and heavy. Okay. Let's decode now. Since you have given me the answers and now you're waiting for me probably to answer. This is a Miesenthal experiment. What was done? First of all, you had grown the E. coli in N15 medium. Okay, N15 medium. And nitrogen is a part of your DNA. So the E. coli took that nitrogen and made the DNA which had what? Which had both of these strands made up of N15. Done. Okay. Now, we are going to grow it in N14 medium. N14 medium. After 20 minutes, the E. coli is going to divide. When the E. coli divides, Baba, the chromosomes which are there, they are going to separate. This is one strand. Okay. Sorry. The DNA which is there, it is going to be one strand over here, another strand over here. This is both going to be N15, N15. But now you're growing it in N14 medium. So the new DNA strand which is going to be made will be what? Will be N14. Agreed? Will be N14. Right? Yes. Now, here, when you did centrifugation, what did we find? When you put this particular thing inside cesium chloride media and you centrifuged it, you found that the DNA was settling somewhere over here. The DNA with N15 was settling somewhere over here. Why? Because it's a heavy isotope. It's a heavy isotope, which is why it is coming and segregating at the end. Now, when you put this particular DNA and you centrifuge it, what do you find? You find that basically they do not go to the bottom. They are somewhere in the middle. Why are they in the middle? Because one strand is N15, another strand is N14. This is lighter one. This is the heavy one. This is lighter strand and this is the heavy strand, which is why you'll see a hybrid band being formed over here. Hybrid band. The question says that after 2 generations, this is just first generation. Now next generation also you have to do. Let's do it. So next generation, again you are going to go in N14 only. What is going to happen? You have to basically do this. You have to separate the strands. One strand is going to be N15, right? And one strand is going to be N14. When you separated this. Similarly, you are going to separate this also. One strand over here is going to be N15, and the other strand which is there, it is going to be N14. You just separate it. Now you are making new strands. It will make new complementary strand out there. So when it makes complementary strand like this, what do you find? This is going to be N14. This is also going to be N14 because you're growing, still growing in N14 medium only, right? This is also going to be N14. This is also going to be N14. What do you see? Two of them, Baba, two of these strands are going to be hybrid strands, which is why when you centrifuge it, you will be able to find what? You will be able to find a hybrid band being formed over here. Right, this is a hybrid band that is being formed over here. You will also find one more band because you find that two new strands are being formed and they are made just of N14, nothing else. So basically there will be a light strand also which is going to be formed over here. So light strand and what? A hybrid strand is going to be there. Agreed? Yes. So now the question is the result. When you are growing this, after 2 generations, the bacteria are isolated from the medium and the DNA of the bacteria centrifuged in cesium chloride. The result of the density gradient of the DNA is going to be what? Is going to be what? Is it this or it is this? It is going to be D. It is going to be D, Baba. The answer is going to be D. Got it? So the children who said C as the answer, I hope you have got it corrected. Please do not do this mistake. It's a very important question. And I have seen multiple questions coming off from this particular thing. Okay? Then, ma'am, can you make related video scope of biomedical engineering? I'll check through it. Okay? Not committing to it. I'll check. Guys, if it is clear, can you give me a thumbs up? Definitely sole entity. We'll be doing that. Ma'am, not understood. Doubt is there. Okay, clear. Listen to me. Rather than me drawing the diagram, I'll show it like this. The same thing is the question. The question says that first you are growing the E. coli in N15 medium. Okay, let's grow it. When in N15 medium, it will be taking N15 isotope in the DNA. N15 isotope is a heavy version. Now, when we put it in cesium chloride to centrifuge it, basically you will find a heavy band out there. Why? Because it is heavy, it will be moving down. Okay, so this is the band that we observe. Now, when we grow the same E. coli in another medium that is having N14, okay, now you are not providing N15, you are providing N14. What is going to happen? The E. coli is going to grow and divide, but it also has to divide its DNA. It has to give it to the progeny also. So what does it do? This particular strand which is there, it will divide and you have 2 new strands, the red ones. They are N15. But the new strands which are going to be formed, the complementary strands which are going to be formed are going to be N14 DNA, right? Because that is the only medium that we are providing. So the new strand is going to be the green one. Agreed. Now when you centrifuge it, do you see a heavy band? No, because you do not have a combination like this. You have a hybrid combination where both light and heavy chains are coming together. That's why you see a hybrid band right over here. This is first generation. 20 minutes means first generation. Every 20 minutes it is going to divide. Now the second generation basically means what happens? Again you are growing in N14 medium itself. You are going to make sure that this particular DNA strand is going to separate. Right, and we have to make a complementary strand. So this strand separate, one red and one green. The new strands which comes along over here are also going to be green because you're still growing in N14 medium itself. The same thing happens over here also. These two guys separate, one red, one green. The new strands are going to be green only, that is N14. Right. Now when you do that, what do you find? When you centrifuge it, you find that 2 strands are there which are still hybrid, which is why you see a hybrid band. And 2 strands are there which are going to be light because they are made of N14. They are made up of what? N14, which is why it will be going to be light in shade. You get it? So you see light and you see hybrid. No heavy chains are going to be seen. Cool? Clear? Clear? If it's clear now? Yes, perfect. Next question. The codons UUU and UUC code for phenylalanine only. This feature is genetic code is called what? Is easy question. Come on. Degenerate codon C. Oh my God, so fast. Sole entity. I have to check dear. Okay. Yes. C. Ranjit says B. Always Shaikh says C. Vidya says C. Viti says C. Sagar says C. Mohammed says C. And Sanjana says C. Manoj, Vidya, Ranjit, everyone. Most of you guys are saying GS World Cup also says. Annabha is also there. Hello. Says A. Okay. Most of you guys are saying C. So answer is C. Remember always when you have multiple codons. Okay, UUC, all of them are coding for what? Phenylalanine. So one amino acid is there, but multiple codons are coding for it. This is degenerate feature. How do you remember it? It's degenerate. It's very simple. Just think that UUU thinks that I am the only one who is coding for phenylalanine. But then UUC is also coding for phenylalanine. That is so bad. That is degenerate, right? So you can remember like that. So multiple codons coding for one amino acid out there. Okay? C is the answer for this particular question. Cool? Yes. Next question, 29th. Match the number of nucleotides of genome of column 1 with the organisms of column 2 and choose the correct options given below. So over here you have got the options. You have got the options over here also. Check the answer and let me know. The action dark, darker, darkest. Oh my god, okay. Guys, if at all you can you give me the answer like this? 29, A, B, C, D. It will be really good. Afan, few more questions, 15 more. Okay. Vidya, Ranjit, Fatima, okay. Sanjana says it is B. Rest of you are saying it is C. Okay. Thank you so much, Always Sheikh and Ranjit, for following what I said. It will be very easy for me. Okay. GS World, Malikarjun, Kavita, most of you guys are saying it's C, and Sanjana says 29, B. Okay. Over here, if at all you look, the easiest one to spot for us is 3.3 into 10 to the power 9 base pairs. This is what the haploid content of human beings. Diploid is 6.6. Okay, diploid is 6.6 into 10 to the power 9 base pairs. That is the diploid content of human beings. Normal cells have it. Gametes, Baba, they will be having half of it. So I can make this go with Q. D goes with Q. D goes with Q. I just have one option, and with that I can find the whole answer. Okay, now 4.6 into 10 to the power 6 base pairs. I'm going ulta, I know that. It is the one for E. coli. So in your NCRT, the maximum is 6.6, that is for man. And then 4.6 into 10 to the power 6 base pair. 4.6 into the power 6, it is for E. coli. Now we are left with two of them. 5386, the minimum number of nucleotides over here according to your NCRT is for 5174 bacteriophage. This name is very big, but actually the guy is having little amount of nucleotides out there. Okay, and now we are left with 502 base pairs. This is for another type of bacteriophage, which is known as bacteriophage lambda. Okay, bacteriophage lambda. So which is why your answer for this question is going to be C. Remember it. Highest is human beings. Then in your NCRT, I'm talking just about your NCRT, okay? Then you have 4.6 into 10 to the power 6 base pairs, E. coli. Then the least is 5386, and that is for what? 5174 bacteriophage. And then in between you have 48,502, that is for bacteriophage lambda. 30th question. Let's go. Let's go ahead. Thank you, guys. Okay. The average length of HNRNA in humans is? The average length of HNRNA in human beings is what? 3,000 base pairs, 2.4 million base pairs, or 1,500 bases, or 500 bases. What do you think is the answer? Swapna says B. Sateri. Hello, Sateri. Hello. I don't know. I'm pronouncing it right or not. A. Okay, some people are saying A, and some people, Hassan is asking, is it B? Okay. What do you think is the answer for this question? It is going to be A. The average length of HNRNA. You have seen genetic code, the features of genetic code, and in that it is said the average length of a gene is what? Of a gene is 3,000 base pairs, right? A gene basically means you're talking about in the DNA a part, right? Same thing is going to be transcribed to form an HNRNA. Same thing, introns, exons, everything will be there. So average length size is 3,000 is being mentioned. So in HNRNA also it is going to be 3,000 itself. So answer is A. Clear? It is A. Did you like this particular question? dun, dun, dun. This came in 2017. I answered fast. Yes, you did. Okay, so children who answered fast, and for children who answered slow, you are doing great. Okay, so let's go. So if E. coli is allowed to grow, now this guys, everyone pay attention to this particular question. Very, very important. Okay, if E. coli is allowed to grow for 40 minutes in a medium containing N15, then the number of N14 containing DNA would be. If E. coli is allowed to grow, think. If E. coli is allowed to grow for 40 minutes in a medium containing N15, then the number of N14 containing DNA would be what? Afan, you will get the PDF of this, dear. Okay? And the video will be still there only. After the live also you can watch it, yeah? Okay, Vidya says it is D. Always Sheikh says 31D. Thank you, child. And okay, Mohammed says it is D. Sagar says it is D. Manoj says it is A. Okay, Sanjana is asking whether it's D. Okay, question is not okay. You didn't understand the question. Clear. I'll tell you. If E. coli, well, there is an E. coli, okay, and this E. coli is grown for 40 minutes in a medium that is having N15. N15, that means N15 isotope of nitrogen is being used in that particular medium. Now they are asking you the number of N14 containing DNA. That means a DNA that basically contains something like this. Both the strands to be N14. What do you think is the answer for this question? What do you think? Obviously, it is going to be A, that is 0, none. How can anything have N14 over here? You're growing in N15, so obviously it will have N15 only, not N14, which is why the answer is going to be A. Did you get it? If yes, just give me a thumbs up. 74 children, goodos. Give me a thumbs up if at all it's clear. Yes, perfect. GS World, Mir Jafar, Afan, Marcia, Amita, Ranjit, Prajwal, Chidambar, and everyone. Suresh. Oh my God, so many children. Give me the correct answer from children whom I am not able to tell the answer. I'm so sorry. Yes. One person. So here we have Ankit sir, guys. And Ankit sir gave me.
Live 4 o'clock.
4 o'clock is there.
Yeah.
You want to say? Come.
So the time is changed because they are going to watch IPL match, no? Today. They were asking me last time.
Okay.
Sir, we have RCB match today. I have to watch and also 4 o'clock we have shifted for all of you, okay? So 4 to whatever time it takes. We'll finish before your toss only. You can come.
The actual reason is he want to watch the IPL match, okay?
No, they all know. I actually... It's not like that. Actually, they wanted me to shift it. Okay?
Okay.
Good morning, everyone. Continue with the bio class. See you today at 4 p.m. with my live.
Okay. So be there for his live as well. And now we'll go ahead. RCB follows. See what ticket. Guys. Come on, sir went. So, sir, in sir's live, we can ask him about which IPL team he supports for sure. Okay? So, yes. Going ahead with the next question that is there, 30-second question. The anticodon found on the tRNA for tryptophan amino acid is Dinkichika. Now this question is going to basically make you go like, oh my God, I do not know the answer for this. The anticodon found on the tRNA for tryptophan amino acid is. What do you think? And let me see if at all there is anyone who can give me the answer for this. RCB. Okay, oh my God. Come on, RCB fans. Give me the answer for this question. Where can I get the PDF? In the WhatsApp channel. Okay. Abhi says it is A. Manoj says it is B. Chidambar also says it is B. Okay. Amita says it is B. Muhammad also says it is B. Ganga A. Shekhar C. Shifa B. Okay. Guys, listen to me very carefully. Over here, the answer is going to be ACC. ACC. Just a second, okay? I did a mistake. Anticodon found. Yeah, it will be ACC. So, in the genetic codon thing, okay, if at all you see this particular thing, you need to understand one very important thing. There are 2 amino acids that just has one codon. One is AUG for methionine, Okay, it does not have, methionine does not have any other codons out there, only AUG is there. Okay. Similarly, tryptophan is also another one which is having UGG. No other codons are there for that also. So tryptophan and methionine, they are basically having single codons. Remember their codons. AUG over here, UGG over there. AUG, UGG. Okay. Now you have been asked about what? The anticodon. You are not being asked about the codon, you have been asked about anticodon. So if UGG is there in the codon, what will be the opposite of it in the anticodon? A, C, C, which is why your answer for this question is going to be A. It is going to be A. Is it clear? Chidambar, not clear? Chidambar says not clear. Okay. So I'll explain it again. Listen to me very carefully. Carefully. Over here, I am saying that you have a list of codons, right? In that, there are two codons out there, or basically two amino acids out there, which are coded by only one codon. Methionine by AUG alone. No other codons code for methionine. Similarly, tryptophan is coded by only UGG and nothing else out there. Clear? Remember their codons. Now, here you have been asked the anticodon found on the tRNA for tryptophan. So, if tryptophan is the amino acid over here and the mRNA has UGG, what is there in the tRNA? That is the question. So, opposite of U will be A, opposite of G will be C, opposite of G will be C. Answer will be ACC. Did you get it now? If yes, 174 is equals to 1 plus 4. Yes, 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 yes. Perfect. That's so nice. Okay. Clear? Perfect. Question number 33. Thank you, guys. Okay. So, yeah. Which of the following sequence of mRNA are required for translation process but are not translated? Are required for translation process to happen but are not translated. What do you think is the answer for this particular question? Should we remember everything? Actually, to be honest, genetic code, yes, if you can, you try to remember it. Okay? Prajwal, I'll give you the answer for that particular question towards the end of this live. Okay? Because I know if at all I say this, it will be problematic now. So, yes. You'll not listen to my... Okay. Untranslated regions, that is D. And everyone else also. Shifa also said it's D. Annappa also says it's D. Okay. Shifan also said it's D. Sachu is there who says it's, what do you call, UTR. Yes. Untranslated regions. Always understand this particular question. That is, basically, when you talk about an mRNA, okay, there are regions before the 5' and after the 3'. These are known as untranslated regions, UTRs. They are there such that the tRNA can come and attach and the whole initiation process can happen smoothly. Nothing happens to the mRNA, everything is stable. So they ensure all of it. So they are definitely required for the process of translation even though they are not getting translated. So answer is UTR. Next question, 34th question. Okay, is it clear, children? Can you give me a thumbs up? I was not even asking for thumbs up since you were saying fast, fast, fast. Is it clear? I do not mind if you're giving me a thumbs up if at all it's clear. I'll be more than happy. Venkatesh, 33D. Perfect, dear. Okay. 34C. Abhi says 34C. Thank you guys for giving thumbs up. When does the lac operon in E. coli become switched on? When does the lac operon in E. coli become switched on? That means it is able to do the transcription. Repressor binds to the operator, RNA polymerase binds to the operator, lactose is present and it binds to the repressor, lactose is present and it binds to the RNA polymerase. It's an easy question, right? Easy question. What do you think is the answer for this? C is the answer. Lactose is present and it binds to the repressor. So everyone who know lac operon, you might be knowing that over here. When repressor protein alone is there, it goes and binds on the operator region, okay, such that if at all I want to transcribe these genes, I'm not able to do that because RNA polymerase, which is right over here, is not able to move. Why? Because there is someone on the operator region. Now, this is known as switched off condition because there is no transcription that is happening over here. On the other hand, if at all there is lactose, whenever the repressor protein sees lactose, is like, Macha, you have come back, come on. So it leaves the operator and comes and joins with the lactose out there, which is why, and basically, if repressor protein is not there over here, that means smoothly transcription can happen. So that is why we say that lactose is acting like an inducer. What is it inducing? It is inducing the process of transcription. It is helping in the process of transcription out there. So when lactose binds to the repressor, that's when we say that the whole process of the lac operon is switched on. Okay, so C is the answer. Clear? If clear, give me a thumbs up. Everyone, if clear, just give me a quick thumbs up. How many questions are there? 34 questions. So I think 45 questions are there in total. Just let me check. Since you guys are asking me total. 50 questions are there. So 35 is done, 15 more. Okay? Where are we? Where are we? Where are we? We are here. Yes. Ma'am, explain one more time. What? This? Okay. So here, why is the answer lactose? You are being asked, when does lac operon become switched on? That is the question. So when does it become switched on? Basically when, over here, the lactose will be coming and attaching on the repressor. That's when repressor is not able to go and bind up there. If repressor is not binding up there, that means transcription can happen. That's when we say it's switched on. On the other hand, if lactose is not there, repressor still binds on the operator, and if it binds on the operator, the transcription process cannot happen. We say it's switched off. Okay, so answer would be lactose is present and it binds to the repressor. When it binds to the repressor, we say that it is switched on condition. Okay. Yes. Next, in which of the following steps in DNA fingerprinting technique are labeled VNTR props used? Are labeled VNTR props used? In which of the process of DNA fingerprinting do you think labeled VNTR props are used? Tell me. During isolation of DNA and during digestion of DNA by restriction endonuclease, or during electrophoresis, or during hybridization? What do you think is the answer for this? Hassan says it is C. Okay. Mano says it is D. Hello Nikhil. Sachu says it is D. And Apsi N says it is D. Okay. And Always Sheikh is also saying it is D. Okay. Sagar. I'll answer. Okay, dear. Vidya says it is C, and Shifan says it is D. Most of you guys, Sagar is also saying it is D, Vidya C. So C and D is where you are getting confused between, is it? What is C? During electrophoresis. And D is what? During hybridization. Listen to me carefully. What do you do over here in DNA fingerprinting? First of all, there's a crime scene over here. Okay, there's a crime that has taken place over here. Now I have a confusion. Okay, police came and police thinks that basically either Jasmine did it or basically Sagar did it. Okay, either Jasmine or Sagar. Who did it? Let's inquire. So from here, I got certain things. Listen, this concept is going to be important for the next question as well. So listen to me carefully. From here, I got some sample. This is what? This is basically hair sample. From the hair sample, basically, I extracted the DNA. I got the DNA. Okay, I got the DNA of the person who probably could have done the crime. Now what did I do? I made a complementary strand of this. Artificially, I made a complementary strand of this and I labeled it with a probe. I labeled it with something else, okay, with a radioactive thing out there. What do you call this thing as now? This particular unique DNA strand which is there, which I have made, okay, and it has radioactive thing attached to it, I will call this as a probe. And now we will see when do you use it. So that Jasmine and Sagar were called. Me and Sagar both were called. And basically our sample was being taken. What was being seen over here when our sample was being taken? Basically, the DNA was extracted and the DNA was extracted from here also. That is the first process, isolation of DNA. Second thing what we do is that we cut the DNA. That is cutting of the DNA or digestion of the DNA by the restriction endonuclease. Okay, ma'am, you cut it, no problem. Okay, that cutting was also done. Just imagine that the cutting is also being done. Now what? Now do you do electrophoresis. In electrophoresis, what exactly happens is that the DNA is going to separate out. Okay, so it's Jasmine ma'am's DNA over here and it is Sagar's DNA over here. It is being separated. Done. Now, can I see anything? No, because it is a gel electrophoresis chamber. What do I need to do? I need to do southern blotting. Okay, I need to transfer whatever DNA is there onto a nitrocellulose sheet. That is known as transferring. So this was a third step. Now there is a fourth step, that is where you transfer the things. Suppose you transfer this onto a sheet. This is the sheet that is there. Okay, again, this is Jasmine ma'am's DNA out there and this is Sagar's. This is on the sheet. Now I am not able to view it on the sheet. So what do? I do one more very important thing. I basically take this particular thing and I make it run on the sheet. Please understand, it will only go and bind to the areas which is complementary. And suppose it comes and binds over here. Suppose it comes and binds over here and I am able to see it. That means that basically the person who did the crime was me. Because it's the same DNA strand, the complementary strand I made from here. That is why it is able to go and bind up there. So I see the same pattern out there. This process where your labeled VNTR probes are going to be used, this is known as hybridization. You are going to hybridize with the labeled VNTR probes and it is only going to bind to the complementary regions out there and it will show the color up there. Do you get it? So I did the crime. Got it? So it's during hybridization. That is your answer for this particular question. And you will also need a UV radiation or basically autoradiography machine to view this. Cool? Yes. Is it clear? Everyone, if it is clear, just give me a thumbs up. Everyone, if at all it is clear, just give me a quick thumbs up. Okay, ma'am. No, I did the crime. So let's go ahead. Question number 36. Match the process of column 1 with the enzymes involved in column 2. Ma'am, why did you murder? I murdered a cheaty, not a human being, okay? Okay. You're under arrest. Okay, guys, tell me the answer for this. Vimla, do you want me to block you? Please do not spam. Please not spam. I have been seeing you from a very long time now. Okay. So Vimla or Vimal or anyone who is behind the name, please do not spam, dear. Here there are children who are attending the session. Let them do that. Yeah, okay. Side ma'am. Okay, sorry. Clear? So C, Manu says C, Annappa says D, okay. And Vidya says A. 36B is what Apsi says, okay. Mohammed says A. And Nikhil, you're most welcome. And, okay. A. So most of you guys are saying A. Let's me try to do it. So DNA replication is done by DNA polymerase. We all know that. So A goes with Q. A goes with Q. I have all the options like that. Now I always say match the following. A and D is the first thing that you have to match. Reverse transcription. So normal transcription is done by basically RNA polymerase. Reverse transcription is done by reverse transcriptase. Okay, reverse transcriptase does reverse transcription. So D goes with R, D goes with R. I just have two options like that. So basically I am going to cut out. The option does not match it. Now translation and transcription is present. Transcription is the easy one. Transcription is done by RNA polymerase. So C goes with P. C goes with P, and here I have got my answer. The answer for this question is going to be B. And what is there left? It is translation. Translation, in that you require amino acyl synthetase. Where do you require amino acyl synthetase, Baba? Listen, over here, when you make the tRNA, remember, you need to attach the amino acid. With the help of the enzyme amino acyl synthetase, basically you are going to do that. That is why we say in translation you require that. Is it clear? So the answer for this particular question is going to be B. Clear, Macha? Ma'am, how to see the previous videos? I answered first. Is it, Hazira? Okay, kudos. You can get, it's there in the, like if at all you go and watch the previous lives out there, it's there in our channel itself, dear. Okay? Perfect. And these questions, PDF, where will we get? In the WhatsApp channel. Make sure you join the WhatsApp channel. After this class, you will get it. Next question. Clear bro. Thank you so much, guys out there. Let's go. According to Human Genome Project, the total number of genes in human genome is estimated at 30,000. The number of genes present on Y chromosomes are. The total genes which are there, 30,000. That is just to deceive you guys. The clear question is, what are the number of genes on the Y chromosome? So answer this question. What are the number of genes on the Y chromosomes? Okay, Vidya says it is B. Cook with a cuckoo is saying it is C. Bharati says it is A. Gagan says it is C. Annappa says it is B. Tasu says, why there's a confusion, guys? C. Hassan says C. Apsi says C. Manoj says C. Sachu says C. Sagar, Muhammad, Shifan, Suresh, GS World, Rekha, Ramurti, Kirti always said. Okay, so multiple people have multiple answers over here. So the answer for this question is 231 genes. C is the answer for this particular question. Okay, and doesn't chromosome 1 has 2968? Yes, exactly. So here you have been asked about chromosome of Y. Chromosome of Y, which is found in the case of males, have the least number of chromosomes, Baba. It is 231. And chromosome number 1, the first chromosome out there, it has the highest number of chromosome, and that is 2968. Remember, the 7 is not there, rest everything, 6, 8, 9, everything is there. So 2968 is found in the case of chromosome number 1, and that has the highest number, and the least is 231 in the case of Y chromosome. Okay? Yes, Gagan, Y chromosome has the least genes. Yes? Clear? Okay. Now the next question that we have, question number 38. Clear, guys? If it's clear, can you quickly give me a thumbs up? And by the way, I hope everyone have clicked on the like button. If not, do that. Okay? Thumbs up. Okay, cool. Let's go. In a crime investigation, the investigating officer collects different biological samples from the crime spot for DNA fingerprinting analysis. Which of the following samples is not helpful in the analysis? That is the question. Not helpful in the analysis. Skin shreds, erythrocytes, semen sample, or hair follicle. What do you think is the answer for this particular question? Tasu says it is B. Vidya says it is C. Shifan says it is B. Okay. Hassan gave me the answer first. It says B. Okay. Chidambar says D. Marcia says it is B. Apsi says it is B. Okay. Lots of children out there having different answers. Okay. The answer for this question is going to be B. Erythrocytes. Skin shreds, skin cells have the DNA. They have the nucleus, everything. Semen sample, semen has the DNA, have the nucleus, everything. Hair follicles have the DNA, have the nucleus, everything out there. Erythrocytes, that is the red blood cells. Red blood cells, Baba, they do not have the nucleus. If no nucleus, no DNA. Initially they have, later on when they mature, they do not have the nucleus out there because they need space to store too much amount of hemoglobin. If DNA is not there, how will you do DNA fingerprinting? Which is why it is not going to be useful. So please understand, blood sample can be used, but in blood RBC is also there. RBC cannot be used. There are multiple other type of cells which can be used, but not RBC. Is that clear? So the answer for this question is going to be B. Okay? Kirti, one of the best teacher out there. Thank you, that's so sweet of you. Okay. If it's clear, everyone give me a quick thumbs up right now. Yo, my back is paining. Okay? Okay. Perfect. Next, question number 39. And it goes like this. Which of the following statements are correct with reference to prokaryotic genome? Again, be careful over here. You have been asked about prokaryotic genome. Monocistronic structural genes, introns absent in structural genes. Transcription and translation are coupled processes. Primary transcript undergoes splicing, and only one RNA polymerase is present. What do you think is the answer for this question? Sachu says the answer is B. Okay. What about others? Hajira says it is C. Okay. Apsi says it is B. B, C, E. Okay. Darshan says it is C. Shifa says it is B. Manoj says it is B. Vidya says it is B. Most of you guys are saying it is B, which is why we need to check it out. Let's check it out together. Come on, guys. So, prokaryotic genome will be having polycystronic nature. That means what polycystronic nature means? They have basically in one unit, they have multiple genes out there. And these genes are going to code for multiple proteins out there. You might have seen lac operon. In that you've seen ZYA. In one structural unit, you have 3 genes, or basically I'll say multiple cistrons out there. This is known as polycystronic nature. So P for prokaryotes as in bacteria, and P for polycystronic. Monocistronic means only one structural genes. It means one gene coding for one protein. This is a unique feature of eukaryotes, not prokaryotes. This is a feature of eukaryotes and not prokaryotes. So monocistronic structural genes is not a feature of prokaryote. Wrong. Here. Next thing that we have is what? Introns absent in structural genes. They do not have introns. Prokaryotes actually do not have introns. So this is correct. Control, Jasmine. Control. Okay. Transcription and translation are coupled processes. That means they happen together. Do they? Yes. Why? Because listen, in the case of eukaryotes, they have cell. Inside the cell there is nucleus. Nucleus is the place where transcription is going to happen. Outside the nucleus is the place where translation is going to happen. So there is no coupled process that is happening in the case of eukaryotes. But in the case of prokaryotes, they are like much chill. We do not have nucleus and all. Transcription, translation, anything can happen together. So basically this is correct. Primary transcript undergoes splicing. What is splicing? Splicing basically means when the DNA, when you make RNA, it will all have introns also. You have to remove the introns. That is process known as splicing. In the case of prokaryotes, you do not have intron. We just said that. So what are you going to remove? You're not going to remove anything. So there is no need of splicing there. So this is wrong. Now only one RNA polymerase is present. Yes, in them. In their case, in the case of prokaryotes, they do not have multiple RNAs or the RNA polymerase 1, 2, and 3. That's for eukaryotes. We are complicated. Prokaryotes are not. They just have one. So basically, can I say that B, C, and E is the correct answer? Yes. So your answer for this question is going to be B. And all of you are doing absolutely great, and I am super proud of you. Good morning, ma'am. Very good morning, dear. It's actually good afternoon now. Okay, so yes. Are we ready for going ahead with the next questions? 10 more questions and then we are done. Then ultimate break is there. You can go have a break. You started doing the next question now. Wait, I was waiting for you to give me thumbs up. Okay. RNA polymerase 2 is responsible for the transcription of. Hassan says 40C is the answer. 40C, okay. Sachu says it is C, okay. Sagar says it is C, Smiley says it is A, and okay. Shifan says it is C, Tasu says C, okay. GS World, Vidya, Sachu, Manoj, Gagan, Ganavila, hello, and Erama, and Ranjit, Ramu, Murthy, and oh my god, so many children are saying the answer for this question is going to be C. Is it C? Yes, it is going to be C, H and RNA. How do we remember it, ma'am? Always remember, we have 3 types of RNA polymerase. RNA Pol 1, RNA Pol 2, RNA Pol 3. Right. The first priority is given to the one that starts with R only. So that is going to be rRNA. That is going to be rRNA. Got it? Yes, ma'am. Okay. In rRNA also you have different types. Anyone remembers the types of rRNA out there? If yes, let me know over here right now itself. Okay. Now, in RNA polymerase 2, what are they going to do? They are going to do the transcription of hnRNA. That is the precursor of mRNA. So from the DNA you get hnRNA, right? So that second priority is given to formation of hnRNA, which is going to later form the mRNA out there. Now, RNA polymerase 3. T is for tRNA, so it is the one that helps in the transcription of tRNA. But apart from that, it also helps in the formation of small nuclear RNA. It also helps in the transcription of some type of rRNA. And what is that some type of rRNA? 5S rRNA. So usually all the different types of rRNA are being made here only, but one type, 5S rRNA, is made over here. And no one told me what are the different types of rRNA that is being formed over here. Hassan, and all the children who were there previously in PU2, can you tell me? Everyone who attended the live previously, can you tell me the different types of RNA being formed over here? I'll be really sad. 5.8S. Okay, one is 5.8S, agreed. One is going to be 28S, exactly, GS World, perfect. So 28S, 18S and 5.8S. Everything ends with 888. Always remember that. Okay, this is also important. Cool? Perfect. GS World, Tasu, Sachu, super proud of you. Okay? And everyone else who is watching this particular video, because you guys are absolutely rocking. 41st question. I hope it's clear to everyone. Children, it's clear? If yes, give me a thumbs up. If yes, give me a thumbs up. Fast. The class will be ending shortly, dear. I just have to go. 40th question we have reached, right? 10 more questions are there. Thank you guys for giving the thumbs up. Okay, let's go. The mRNA has untranslated regions, UTRs. Where do they have it? At the 3' end beyond the terminator codon, at the 5' end before AUG, that is the start codon, at both 3' end and 5' end beyond the terminator and before the AUG respectively, and AUG and terminator codon flanks the UTR. What do you think is the answer for this question? Hello, Kid Zone. Manoj says the answer is C. Fax says the answer is C. Shifa says it is C. Tasu says it is C. Sachu also says it is C. Priyanka, Eramma, everyone says it is C. Always Sheikh, Ranjit, Mohammed, Atif and Hazira, everyone, perfect. Kids Zone, everyone, cool. The answer is going to be C. I told you it's going to be before 5' end, that is this region, and after 3' end, that is this particular region. So this is AUG over here, and here you will have stop codon. So please know all these things. So AUG, initiator codon, will be over here. So don't be confused. They can give you 3' what do you call, before the UGA respectively or UAG respectively. So don't get confused with all of that. Please know these things, which they are super important. Okay? Yes. Next, 42nd question. Histone proteins are positively charged because they are rich in basic amino acid residues such as what? Histone proteins are positively charged because they are rich in basic amino acid residues such as arginine and lysine, arginine and phenylalanine, arginine and proline, and arginine and alanine. What do you think is the answer for this question? Abba, so much fast answer. Ma'am, please answer my question. What is your question, dear? Tasme, it will be ending up soon, dear. Okay. A. Most of you guys are saying it is A. And I'll just read out a few names. Darshan, Kirti, and Viti, GS World, Facts, Always Sheikh, and Vidya, Hazira, Tasu, Sagar, Kid Zone. Okay, so the answer is going to be A itself, arginine and lysine. So always remember, you have your histone proteins. On the histone proteins, if at all you want the DNA to be wrapping around, and DNA is negatively charged, that means it has to be opposite. Opposites are the one that attracts. So DNA is negatively charged and your proteins are going to be positively charged. Always remember, it is Los Angeles, lysine and arginine. Lysine and arginine, do not get it wrong. Okay, cool. Yes, yes. Next question. Why it is 41st question again? Just a second. So I have repeated a question. So that means you have got extra one question. Okay. During transcription, the DNA strand with 3' to 5' polarity or structural gene always acts as template because what? Because of what reason? During transcription, the DNA strands which is in this particular polarity, that is 3' to 5', is the one that always acts as template. This is the one that undergoes transcription. Why? Ma'am, I have completed one chapter of math of PU1. Now 3 chapters are there for 2 subjects. Can you tell? Okay, Sagar, can I tell at the end? Because I did not, I need to understand it. Okay? D. D is what? Enzyme DNA dependent RNA polymerase always catalyzes the polymerization in 5' to 3' direction. Exactly. So here you are going to need the help of RNA polymerase, and it will always basically make a new strand in 5' to 3' direction. So why not the other options? Because here it is written enzyme DNA dependent RNA polymerase always catalyzes in 3' to 5'. Wrong. It is in 5' to 3'. Enzyme DNA dependent RNA polymerase always catalyzes polymerization in both directions. Again wrong. Okay, now see, nucleotides of DNA strand with 5' to 3' are transferred to mRNA. Does not make any sense, which is why you only have one answer, that is D. And always remember, they can confuse you. They can put RNA dependent RNA polymerase. So please be sure that you read the question carefully, whether it's DNA or not. So which is why your answer for this question is going to be D. Is it clear to everyone out there? Yes? Cool? If yes, give me a thumbs up. No. Okay. It is not clear. Ma'am, when will the next class start and which? It will be math and at 4 o'clock. Okay. So over here, when you're saying that this is a strand of DNA, and when you're saying it's in 3' to 5' direction, okay, you will need someone to transcribe it. Who is that? RNA polymerase. It is going to transcribe in which direction? 5' to 3'. It is an RNA polymerase which is dependent on DNA's information to transcribe it, which is why enzyme DNA dependent RNA polymerase always catalyzes polymerization 5' through 3' direction. That is why it needs an opposite strand out there with 3' to 5' polarity. Got it? Yes. And the next question. The number of codons effective in coding 20 amino acids are. The number of codons, okay, the number of codons effective in coding 20 amino acids are. Tasmia, because ma'am today married. Oh my god, Tasmia, you should be going for that. Go, 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 go. 42. Ma'am, if you study Jeevit publication, can we score above 120? You're talking about PU2? If you're talking about PU2, Jeevit can help you, but please refer to some extra questions. B. Most of you guys are saying B is the answer for this particular question. Is it B? The number. Some people have said A also. Some people have said A as well. Listen to me very carefully. Okay. 42, 61 and 3 for stop codons. Exactly. The number of codons effective in coding 20 amino acids are what? 61. Remember, we have altogether 64 codons, but not all of them are coding for amino acids. 61 of them are going to code for amino acids, and basically 3 codons which are there, they are not going to code for anything. They are stop codons. And what do you call them? What do you call the stop codons as? Anyone? Do you know? What do you call the stop codons as? Nonsense. Nonsense. Exactly. Guys, what do you call the, what is the codons which are acting as stop codons? I meant to ask that. U. Okay. Guys, U are. Stop codons. UAA, UAG, UGA. Exactly fact. Exactly. UAG. Okay. It is UAG. Che. Sorry. Where is it? UAG, UGA and UA. UAG, UGA and UA. These are the 3 stop codons out there. Do not forget them. This is very, very, very important. Okay? Terminator codons is also what you call them. There is another name for it. Exactly. Witty. Perfect. Let's go with the next question that is there, 43rd question. Following representations P, Q and R denote few steps of Griffith experiment. Identify the correct ones. Okay. So over here you have got certain steps out there. You have to find which one of them is correct. R strain you're injecting into the mice and the mice dies. Is it? Is it? Yes, exactly, Aapsi. Exactly, everyone who gave me the answer for that question. You guys are doing absolutely great. Ma'am, the question from KCET comes from this video or not? This is PYQ. Obviously, you might get repeated concepts coming up there. This video is important. You can watch it whenever you have time. If you are not able to watch it now, Tasmia. Okay? Yes. A. Fact says the answer is A. Anappa says it is D. Okay. Cook with Cuckoo is saying it is D. Chidambar is saying it is C. Okay. Hassan says only R is true. Multiple answers are there. Let's go ahead. So R strain, when you're injecting into the mice will not die because R strain is a rough strain. It is not virulent. So this particular statement is wrong. Now Q. S strain, heat killed. Inject into the mice and the mice die. S strain is actually virulent, but this is heat killed, so it will not show the effect, which is why mice will also not die. Papa mice is now free to live. Okay. Now R strain you're injecting into the mice and the mice lives. R strain, if at all, you are injecting into the mice and the mice is living. Is that true? Definitely yes, because this is non-virulent. So can I say that only the R statement is true? That means that B is your answer for the question. Simple. Clear? Clear, clear, clear. Okay? Give me a thumbs up if at all it's all clear. And everyone did absolutely good. Sagar, Viti, Priyanka, Chitambar, Ramurthy, Amita and Vijay. And Samrina, I already gave you the answer here. GS World, Kids Zone, Amita, Vijay Tales, Anappa, everyone. Apsi, Swapna, Sudeep, Nikhil, Kids Zone, Anshul, Kirtij and everyone. Rutika, Hazira. By the way, if at all I have missed any names, I'm so sorry. Okay? Yes. Next thing. Question number 44. In structural genes, the template DNA strand has nucleotide sequence that is this. Very careful. Okay, the template DNA strand has a sequence like this. Find the correct and complementary nucleotide sequence on the coding strand. On the coding strand. Give me the answer for this. Priyanka, okay. Priyanka, do you mind if I do it at the end of the class? There's 6 more questions only left. I'll do it, okay? I promise. You just note down the question number. We'll come back to that. Okay. So 44C, Muhammad says it is C, Vijay says it is C, Hassan says it is C, okay. Anapa also said, Anapa has a different answer over here, Rutika also has a different answer, okay. But most of you guys are saying it is C, and C is what? C is this. Why do you say it is C? Listen, if this is template strand, this has to be the coding strand. Template strand is always in 3' to 5' direction. The coding will be always in 5' to 3' direction. Whatever thing is there over here, for example, when I say that it is A, T, G over here, the complementary strand will be over here. That means it will be similarly T, A, C over here. That is the logic. So when I'm saying that this is the thing that is there, so over here also you will be having TAC, and the list goes on. You have to make sure it is from 5' to 3' also. So here, my dear children, when you see the sequence, there are 2 of them which are complementary, but one is in 3' to 5', other one is in 5' to 3'. You have to know this, please remember it, and the answer is C for this particular question. Okay, clear? Is this question clear? If yes, give me a quick thumbs up. Quick thumbs up. Perfect. Thank you. Okay. Next question. Experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on which plant? Experiments involving use of radioactive thymidine. You know what? This particular thing comes under semi-conservative replication. So, Mieselson and Stahl did their semi-conservative replication experiment with N15 isotope, N14 isotope. Similarly, Taylor and other guys out there, his friends, basically they did the experiment using radioactive thymidine. You're being asked that which is that particular plant that was being used for it. Any idea? Okay, oh my God. Okay, trust me on full power. C says C is the answer for this question. Okay, what about others? Okay, 43C is okay. Wait, Sagar says it is B. Rutika says it is B. Sudeep says it is A. Always Sheikh says it is A. Okay, I'm going to read out the name who are going to give me correct answer now. Amita, Vijay, and Anshu. Okay, and Mohammad Zubair and sorry. Guys, the answer for this particular question is going to be A, Vishya faba. Faba beans. You also call this as faba beans. And if at all you have no idea about how it looks like this. Have you had it? It is Vishya faba or faba beans. Please remember it was radioactive thymidine. This is also important. And who was doing the experiment? It was Taylor and his colleagues. Thymidine was the thing which was being used. Is this clear? Apsi, yes, 45A, Taylor has done that. Exactly. Clear? If clear, give me a thumbs up. It is not C, it is A. Okay? Thank you, guys. Let's go ahead. Now we are entering the zone of new questions, no PYQs. Okay, 5 questions will be there and they are new questions. Make sure you pay attention. It might be tough, it might be easy also. So, what will be the percentage of guanine in a DNA molecule having 20% of adenine? What will be the percentage of guanine in a DNA molecule having 20% of adenine? Okay, 46B. Renuka says B. Hazir says B. Okay, Always Sheikh said already. Okay, Hassan says 20%. Okay, most of you guys are saying it is B. B is what? B is 30%. It is it? Let's check it out. So what is said is that you have adenine to be 20%. That means thymine which is there, it will also be 20%. Why? Because A base pairs with T, right? Okay. What about guanine and cytosine? Listen, if I'm talking about percentage, the highest percentage is going to be 100. Out of 100, 20 plus 20 is been already covered by these two guys. So 40 is been done. What is left? 60 is left. Now 60 we have to divide between both of them. So here it will be 30 here it will be 30. So guanine is going to be 30%. Always remember, here they have not mentioned anything else. Sometimes they can tell in a single stranded DNA thing how it is. Guanine is this much, how much adenine is there? It will be 0. In single stranded, how can you guess? A might be multiple, T might be multiple. There is no base pairing happening, right? So if at all single stranded is not been mentioned, just assume it to be double stranded itself and give your answer. But if a single stranded is mentioned, please be very sure you do not give wrong answer. Okay, we cannot tell the percentage, it will be 0. Cool? Is it clear? Macha, it's clear? If it's clear, give me thumbs up, everyone. Okay. Ma'am, speed. What okay. 47th question. In Hershey-Chase experiment, when radioactive viruses were allowed to infect E. coli bacteria, what was observed? When radioactive viruses were allowed to infect E. coli, what was being observed? What is the answer for this question? E. coli, which were infected by viruses with radioactive DNA, became radioactive. Did they become radioactive? The viruses which had radioactive DNA, they transferred the DNA to the E. coli, which is why it also became radioactive. That is actually correct. Correct. E. coli, which were infected by viruses with radioactive protein, did not become radioactive? Yes, because radioactive protein was a part of what? The capsid layer, the outer layer out there, which is why the E. coli, which was infected with the radioactive protein thing, it was not becoming radioactive. That is also correct. E. coli, which were infected by viruses with radioactive DNA, did not become radioactive. That will be wrong because it became radioactive. It passed the radioactive thing. E. coli, which were infected by viruses with radioactive protein, became radioactive. That will be wrong to say. So only answer for this question is going to be what? 1 and 2. Your answer for this question is going to be A. It is going to be A. Got it? And every one of you gave me the answer as well. And I'm super, super, super proud of you guys. Clear? If clear, give me a thumbs up. Thank you. Okay, perfect. So we are going ahead with the next question. And it goes like this. Following statements for mRNA maturation are given, marked as 1, 2, 3 and 4. Okay, what do you have to do, dear children? Select the correct option. So we have to find which one is correct. Okay, capping of methyl G occurs at the 5' end. This is for mRNA maturation, as in when you had hnRNA, you are going to do splicing, you're going to do capping, tailing and everything, and finally you're going to get the mRNA. And the processes are being asked over here. Does capping of methyl G occurs at the 5' end? So this is the 5' end and this is the 3' end. Do you think capping occurs over here at the 5' end? Yes, it does. Capping of poly AAA tail occurs at the 5' end. No, that is tailing, that is not capping. Okay. Tailing of AAA occurs at the 3' end. Does it happen? Yes. So listen to me carefully. At the 5' end, you have methyl G group being added up. At the 3' end, you have multiple adenine group being added up. This is known as tailing. This is known as tailing. Why are we doing all this thing? We want to protect our mRNA. Our mRNA was inside the nucleus. It was a papa guy, and now we are letting it to go explore. When it is going to explore out of the nucleus, there are monsters out there which might probably cleave it. And we don't want that to happen. That is why we do capping and tailing. Capping at the 5' end with the help of methyl G, and tailing at the 3' end with the help of polyadenylate tails. Got it? So your answer for this question is going to be what? 1 and 3. 1 and 3. 1 and 3. That means B is going to be the answer. Is it clear? Yes, perfect. All of you have given me the answers. Is it clear, children? Dreams, I'll take your question towards the end, okay? Just give me some time. Yes, ma'am. Perfect. Okay. 49th question goes like this. So over here, yes. The given figure represents the process of transcription in bacteria. Select the option which correctly labels as A, B and C. So over here, this is A, this is B, and this is going to be C. Okay, so now what do you have to say? You have to choose the correct options out there. Let's go. what is going to be A? So this is your DNA. From the DNA, something is coming out. What is it? It is going to be the mRNA for sure. So A is going to be mRNA. Okay. What about B and C? This is the problematic thing that you mostly face. What is B and C? B is going to be your RNA polymerase, and attaching to it, what do you have? You have the rho factor. Rho factor is the one that will be helping in termination. That is why your mRNA is separated now. Okay, so always remember that. So your answer for this question is going to be B. A is mRNA, B is RNA polymerase, and C is the rho factor. The one that helps in attaching to the promoter is sigma factor. Remember, RNA polymerase is too shy to go and attach, which is why it needs sigma factor. S for shy and S for sigma. And at the end, it is crying because it is what you call breaking up with the mRNA and DNA are breaking up. That is why rho factor. Okay, remember that. Yes. Is it clear? Finally, we are going to go ahead with the last question. So thumbs up needed. Thumbs up. Yes. Okay, so it goes like this. In the given figure of lac operon, an operon for inducible enzymes, identify components and the enzymes. Over here, you have the components, and I hope it's visible to you. This is X. Onto X, something is coming and attaching, that is Y. This is E1, this is E2, and this is E3. Basically, Z codes for E1, Y codes for E2, and A codes for E3. You have to tell me what is Z, Y, and you have to go like that. And you have the options like this. You can choose from the options. 45. 45. Guys, give me the answer. Last question. 50. I'll read out everyone's name. Okay? So, fact said it is B. Okay. And Hassan, okay. Sudeep says it is D. And Vijay says it is C. Dreams Edit says it is B. Shifan says it is B. Vijay C. And Muhammad Atif B. Samarth, okay. Vijay says C. Sudeep Police D. Sidhu says thank you. Okay. Sachu D. Muhammad Amita says C. Sahana says B. Manish says D. Allah Baksh says C. Always Sheikh says C. Kirti says B. Venkatesh says C. Suvarna says D. Hazir says B. And Sanjana says A. Sidhu says D. Kagan says D. GS World says C. G. Tilak says D. Ramamurthy says B. Ritika says C. And Manish says D. Mallikarjun says B. Any name I have missed? I hope not. Okay, Mr. Sajan is also there. So Kid Zone, Apsi, everyone, Vinita, Hanu, thank you so much for attempting the question. The answer for that particular question is going to be D. Why D? X is going to be the repressor protein. Look at this. X is a repressor protein onto which Y is going to come and bind. Y means what? It's lactose or allolactose. Over here, inducer, that is given lactose. Done, done. Now, E1 is going to be beta-galactosidase. E2 is going to be permease. And E3 is going to be, last one, transacetylase. So, beta-galactosidase, permease, and transacetylase. And the answer for this question is going to be D. Clear? If clear, give me a thumbs up. Bye, ma'am. Catch you with the next class soon. Definitely, Hassan. Bye-bye. So those who have doubt can stay back, and everyone else can leave. And thank you so much. Before leaving, make sure you hit on the like button as well. And be there for the next live. Yeah? Thank you so much for being there. I truly appreciate your efforts. For everyone who have been there from the starting to the end, you guys are doing great. Please keep on being consistent with this. You guys will rock your KCET examination, and I pray for that. Wishing you all the best. Lots of love to you. Okay? You're most welcome, Kirti and Venkatesh. Next ka hai ma'am class. So class is going to be there on Monday. Yes. On Monday. Okay? Bye-bye, bye-bye, everyone out there. Okay. Now, Priyanka, that mice die question, please explain. Mice die question. Can you tell me the question number? I might have a picture, I might not have. If you can give me the question number, it would be really good. Question number, every day. Mice die question, we did it recently, right? Yeah. Okay. Which chapters? I think I'm doing human health and diseases and some other chapter. I have to check once. Thank you, ma'am, for this productive session. Thank you, Apsi. Okay, so those of you who were here, listen to me. This was the question that you were confused for, Priyanka. Griffith experiment, right? When R strain is injected in the mice does not die, because R strain is what? There are two strains. One is R strain, one is S strain. R strain is a rough strain. Why is it rough? Basically, it does not have a polysaccharide covering, and since it is not having a polysaccharide covering, we say it is non-virulent. Okay, it does not cause any trouble. S strain, Baba, it has a smooth covering, looks very smooth. Why? And Baba, it has what? It has a polysaccharide covering, that is why it looks smooth, but it is virulent. It is the one that is virulent. It can kill the organism out there. Both these strains have Streptococcus pneumoniae. Now, R strain, when I inject into the mice should not die, it should live. It's wrong because it is non-virulent, right? Now, S strain, when I am injecting to the mice die. S strain, when you inject in the mice will die. But here you have mentioned one more thing, heat killed. That means you're treating it with heat. If you're treating it with heat, Baba, what is going to happen? The DNA is going to be altered. That is why S strain will not do its normal function. That is why it should not be killing. The mice will still live. If at all, it is heat killed, S strain. Now, R strain, when you're injecting into the mice, do you think the mice will live? Obviously, yes. It is going to live. So, can I say only out of 3 options, only this one is correct? So, your answer would be B. Okay, your answer would be B. Okay, just a second, guys. 39 question, I will be taking it up. Okay, wait just a second. Sagar, ma'am, I have completed 1 chapter of maths of PU1. Now 3 chapters are there of 2 subjects according to phase 1. You. Listen. You can divide your time into how many subjects you want to cover in a day. Prioritize the order. Suppose in the morning, you can give yourself 2 slots. In the morning, you can wake up early. You can give 2 slots to yourself where you do one subject once and the second subject second. In that way, in the afternoon also, you have 2 slots for doing 2 subjects. You can do like that. If you feel, I do not understand the question exactly, but I am telling you, generally, if you feel that, okay, I do not want to do 4 subjects, I just want to do 2 subjects. Okay, then please take a required amount of time, whatever time is required for one particular chapter in the first half. In the same time, in before afternoon, try to cover the next chapter as well. At the earliest, listen, for PU1 chapters, you should not be spending too much amount of your time. It will be a foolishness to spend that much amount of your time for your PU1 chapters. So wrap it ASAP. Watch a one-shot video, wrap it as soon as possible, solve the questions. 2.5 hours would be, basically 2.5 hours should be the maximum that you should be spending for that particular chapter out there. Otherwise, it will be a big trouble. And only the chapters which are of high priority. If the chapter is not of high priority, take lesser time. Okay? Yes. Now, okay, 39th question that I'm going to take it up. Just a second. 39th question was this. Where is it? Yeah. So, which of the following statements are correct with reference to prokaryotic genome? Listen, in the case of prokaryotes, first thing that you need to know is that when I say that this is a structural gene can contain multiple cistrons, means units that code for proteins. So you might have seen in lac Operon, ZYA is there. This is one gene only, but it has multiple cistrons. It is coding for multiple proteins. 1, 2, 3 proteins are being coded out there with one particular structural gene. This is known as polycistronic arrangement. But here it is written, monocistronic structural genes are present in prokaryotes. Wrong. In the case of eukaryotes, we have monocistron. That means when you talk about one structural gene, that means there will be only one gene. And this one gene will code for only one protein. This is monocistronic gene that is found in eukaryotes, not in prokaryotes. So that is why this is wrong. Now, introns absence in the structural gene. In the case of prokaryotes, so usually what happens, in our DNA there is introns, there is exons. Introns means the region that do not code for any amino acid. And exons means the region that codes for amino acids. So whenever transcription happens, whatever is there in the DNA, that is transcribed to form an hnRNA. And now, when the hnRNA also have introns and exons, that is a problem. Because this hnRNA will later code for mRNA, and the mRNA is the one that makes the protein. But for formation of protein, you do not need introns. So basically, you have to cut it out. So basically, it undergoes splicing. It undergoes splicing, that means removal of introns, and that is how you get a mature RNA, mRNA, which just has exons. This is a case of what? Eukaryotes. Prokaryotes do not have introns at all. Introns are absent in the structural genes. This is correct for prokaryotes. Now, here I'll go with the next one first before going with this. Primary transcript undergoes splicing. Splicing means removal of introns. If it does not have introns, do you think it will undergo splicing? There is no requirement for that. So I'll say this is also wrong. So what are we left with? C option. Transcription and translation are coupled processes. Listen to me carefully. When you talk about a prokaryote, it does not have nucleus and cytoplasm and things like that. Everything is arranged in a similar region. Whereas when you talk about a eukaryote, what do you have? You have a nucleus where transcription is going to happen. You have cytoplasm where translation is going to happen. So we say it's not coupled. Both of them are not going to occur together. But over here, since there is no segregation, everything can happen in a coupled way, in a simultaneous way. So I'll say this is correct. Now, only one RNA polymerase is present. For transcription of DNA to RNA, in the case of prokaryotes, you just need one RNA. That is absolutely true. So this particular statement is also correct. So can I say B, C and E is correct? B, C and E is correct, which is why we say the answer for this particular question is going to be B. Is it clear to you, Anusha? Is it clear to you, Anusha? Sorry, Anshu. Okay? Now, in Drosophila, gene A and B are linked, okay? The heterozygous female is test cropped with double recessive male. Okay. Dreams edits, I am not able to understand. I might have to see the question. If at all it is there in some book, you can tell me. I can solve it and give it to you later on. Okay. Okay, 41st, someone was asking 47th question. Sarjan was asking me 47th question. Dreams edits, please tell me the name of the book and the question number. I can try to find it out, okay? 47th question is what, dear? 47th question. 47th question is this. Okay. Hershey and Chase experiment. In Hershey and Chase experiment, what was being done? Listen to me carefully. There were two bacteriophage. Okay. One bacteriophage, you had labeled it with S35, sulfur 35. Listen, this isotope of sulfur is basically, sulfur is a part of protein, so it will go up and get accumulated wherever protein is made. So the capsid layer, the outer layer of the bacteriophage is the one that has the protein. So the DNA which is there, it does not have the protein, only the outer layer will be having the protein out there, right? Yeah. So what is radioactive over here? The radioactive thing over here is a protein. Cool. On the other hand, here you are labeling with P32. And phosphorus is a part of what? Your DNA. So your DNA is going to get labeled. What is going to get labeled? DNA. Now we are checking what is going to go inside the E. coli. That is the question. So when you treat both of them with E. coli, what is going to happen is that bacteriophage is going to pass down its genetic material, that is the DNA. Here also it passes down the genetic material, right? Just a second, I'll make it in white. But is it radioactive? No, because we put sulfur over here. Here the radioactive thing, the only thing that is radioactive is protein, not the DNA. But here it is the DNA which is being radioactive. Clear? Now you do not know it. How do you know it? Basically you centrifuge it. Later on you get to understand that basically in the centrifuge table you found that there is pellet over here. Pellet means E. coli which is settling down. It is not radioactive. No radioactivity is seen over here. Because the E. coli does not have anything radioactive. We do not transfer the protein, right? That's why. Whereas the supernatant which was there, that was radioactive. Why? Because the proteins which are there, they are left behind. Only the genetic material is passed down. Only the protein is left behind, and that remains. So in the centrifugation thing, E. coli settles down, whatever is left behind stays over there, and that becomes radioactive. On the other hand, when you check over here, you will find that at the bottom, the pellet is radioactive. That means E. coli is radioactive. How can that be radioactive? I have tagged it with phosphorus over here, and it is going to connect with my DNA. That means DNA was being passed down to the E. coli. That is why radioactivity is seen over here. Whereas here in the supernatant, there is no radioactivity because the protein is not being tagged. Now the question over here is, E. coli which were infected by virus with radioactive DNA became radioactive. E. coli which was infected with viruses which had radioactive DNA, do you think it is radioactive? Yes, it is radioactive. E. coli which were infected by viruses with radioactive protein did not become radioactive. E. coli which was tagged with radioactive protein, did it become radioactive? No, it was not radioactive at all. The supernatant was, it was not. So these two statements are going to be correct. 1 and 2 are going to be correct. Okay, for this particular question. Is this clear? I know it's a complicated thing, but yes. Is it clear? Ma'am, these classes are useful for NEET. Listen, for NEET, you require to do NEET PYQs, that is separate, but yes, it can help in some ways. Okay? Clear. So I'll end up the live, guys. Okay, K or Arvind, hello dear. So yes. Cool. DNA fingerprinting forensik question will come or not? It can come, it may not come. Had your lunch? I did not have yet. I need to have it. Okay. So thank you for asking, and just give me a second. Ma'am, thank you for this class. You're most welcome, champ. Thanks a lot for being a part of this particular class. And yes, over here, just give me a quick second. Yes. Okay. So guys, that's all for now. Okay. Lots of love, Priyanka, and everyone out there. Thank you so much. Well, with the next live. Next live will be there on Monday, hopefully. And KR Arvind, I love your class. Thank you so much. Lots of love to you too. And guys, please take care of yourself, and please go make sure that you practice even more questions out there whenever you have got time. And do not waste your time much, okay? Take rest and enjoy your life. Lots of love from Jasmine ma'am, signing off. And do not forget to hit the subscribe button as well if you are someone who have not hit yet. Lots of love. Bye-bye. Most welcome. Thank you. Tomorrow you will be having chemistry and physics class. Okay? Darshan Gowda, when class? Monday. My class will be next on Monday. Bye-bye.